Good morning from the RV Falkor, the Schmidt Ocean Institute research vessel Falkor, on our third expedition to the Great Barrier Reef and Coral Sea. This is Dr Robin Beaman from James Cook University, welcoming people watching in on live YouTube and Facebook. This expedition is the northern depths of the Great Barrier Reef. We've been out here for about a week now, and today we're diving on a very interesting feature. We're calling it the Tydeman Knoll. This is a unusual knoll-like feature about 6 kilometers out from the edge of the Great Barrier Reef. We're in depths of about 550 meters, just on the edge of this knoll. And as we do it in all of our ROV dives, we want to get a representative sample of the seafloor. So we're just taking a push core here, and we're joined by some animals. We've got some squat lobsters, a very unusual looking gastropod snail. It's live on the seafloor right next to the push core, and hermit crabs and a few fish. So we've been on the bottom now for about 5 or 10 minutes, just getting ourselves settled, and we're taking our first push core sample. And there's an inset view looking at 3D mapping that's been conducted over the last week, and just looking from, I guess, a deep fish perspective. The depths you're looking at there is probably from around 2,000 meters deep, so that's 2 kilometers deep, looking up onto the edge of the Great Barrier Reef. And you can see it's very, very heavily covered in canyons, and there is this quite strange knoll-like feature, like a big hill that sticks out from the edge of the Great Barrier Reef. And we're diving, today we're going to be diving up the eastern face of it. It almost looks knife-like. It's about 300 meters in height, that sort of sheer face. And we're starting at the very base of it, and we'll traverse along the eastern face and up along towards the summit and see what we can find.
Anyways, fine, Jared.
So we're just getting our second push core here, and on the ROV controls we've got J-Rod, assisted by Brody, with John and James managing the IT wireless. And also science team on board. We've got Marty McNeil here with me from Queensland University Technology, and Vicky Lowe from University of Queensland, and Joan and Valerie as postgrad students from James Cook University. So a smaller science team on board, but we're assisted by lots of principal investigators watching from ashore, and of course all of yourselves, experts and general public alike, watching on YouTube and Facebook. We really appreciate all the help we're getting from ashore here. So just getting the sit cam view. That's we've just swapped the cameras around. Of course, the ROV itself has got, I think, over 10 cameras. Is that right? 10 cameras, right, at all different angles. So we've basically got a full 360-degree view of what's happening around us. The main camera you're looking at is the science camera. This is the 4K vision. So it's really high resolution imagery. Of course, it's compressed a bit once it goes out onto YouTube and Facebook. But yeah, it's quite amazing to sit here in the control room on board the Falkor. Oh, we've got looks like some crab coming into view here, guys. Common and hot. Yeah, straight towards us. As soon as the sediment clears. Okay, quite a bit muddy, of course. This seems to be a real feature of the deep ocean here. Now, our previous dive, 396, was down the Rotter Canyon. It was quite deep, around 1,700 metres. It was very, very muddy. Here, again, mud seems to be the feature, but it looks a lot more white compared to where we were the previous dive. So it's a bit of an exploration dive, this one, to try and understand what this big knoll-like feature is. Very unusual that it's sitting out from the edge of the Great Barrier Reef, because all around us was surrounded by canyons, but yet there was this very large broad ridge extending out from Tydeman Reef, which is the closest shallow coral reef to us at the moment. And sitting on top of it is this about 300 metre high knife-like knoll feature. So we really want to know what it is, what the geology of it is, and of course what marine life is living in and around on it.
Couple ends sticking up.
Okay, so as the sediment clears, we're getting a clearer look at the seafloor. Jared, could we have a closer look at the these coral colonies? Yeah,
Yeah.
So we have a permit to collect anthozoans and other invertebrates in this part of the Great Barrier Reef. Great Barrier Reef is a World Heritage Area, and it's zoned into multiple use zones, with some zones more protected than others. Classic green zones are no-take areas. And our permit, this area here is outside of a green zone. We have a permit to collect targeted soft and hard coral. So for our biologists ashore, we're having a closer look at some of these coral colonies to see whether we wish to collect them. So the dive today is starting at the base of the knoll on the broad plateau, the broad ridge which surrounds it. So we're down at 540 metres. The knoll itself, the base of it, is around 460 metres. So the idea is that we're going to traverse along the shoulder of this ridge to the base of the knoll, and then we're going to climb up. It'll be much steeper than what we're seeing at the moment. So we're just communicating with our biologists ashore through Slack. We're direct communication with them, and we can, oh, it's just pulled itself back into a hole there. That was interesting. Have you seen that before?
Never.
Whoa. They're supposed to be sessile, not mobile, right? What on earth? Disappearing coral. We can't explain that one. All right, I guess we're not going to be collecting that one. Okay, so we got some sea cucumbers in front of us here. We're just having a bit of a look around. Yeah. Is that a little fish? Something bright. We've seen a few fish already. Yeah, so we're on this. Yeah, it is fish. So we're on the. Or is it shrimp? It's a very, very clear looking shrimp. So we're just on the edges of this knoll, on the flat ridge which surrounds it, and it's clearly muddy, but there's plenty of action down the seafloor. There's a brittle star having a bit of a walk. Yeah.
It's in a hurry.
Beautiful colours. Can you put the lasers on, guys, just so we can get a sense of scale? There we go. A laser's on the bottom centre. So they're 10 centimetres across. Don't know. Just trying to guess what this animal is at the top right here. We've got two brittle stars right in front of us, but at the top right we've got something hanging out of a hole. It has that hagfish look about it, doesn't it?
Is it?
Cucumber? Ah, or an eel. Are you able to just go and touch it, guys? Just see if it... Is it? We might just manoeuvre the ROV.
Yeah. We should go back to that other sea pen slash...
There's another one.
There's another one.
Oh, you want to go to that one?
Yeah. Where was it?
Oh, look, the sea cucumber just lifting up.
Yeah.
Oh, that's, it's a sort of classic escape behaviour. So we've got feather star, lots of brittle stars. There's plenty of life.
Nice little seascape, isn't it?
It is nice. Yeah. More brittle stars over here. Possibly a hermit crab or something up the top. Big anemone over on the right. Is this going to suck into the mud like the other one? That was pretty weird. Was not expecting that.
No. I mean, you expect them to retract their tentacles.
Yeah, but pull it all the way in. Yeah.
No, not the whole stem like that.
So we think this is an octocoral, but what's it attached to? That's the big question.
You want to try and sample it? And if we sample it, is it one for the quiver or
no, just
in
for
a bio box? Okay, we'll try and sample it.
Yeah, so there's we've got a sort of interesting sampling regime happening here where we're working with Smithsonian Institute and the Sanger Foundation to do whole genome DNA sampling on certain samples. They have to be preserved separately, not dropped in a bio box, but put into what we call a quiver, which is just like a big cylindrical tube. So they're completely isolated, from any other samples, and then once they're brought back on board, they are flash frozen in liquid nitrogen. So we have a dewar or a big bottle. Oh, there it goes. What is doing this?
Sure.
That'll keep it cold.
It's not going to go in the liquid nitrogen anyway, but yes.
Well, that's pretty strange. It's the first soft coral I've seen that jumps away from you as soon as you touch it. Oh, I don't know. What is it like? Could it be a polychaete with some sort of a colony attached to it?
Are we sure it isn't some sort of weird sea pen? We're not sure of anything.
Yeah.
Right now.
Yeah. I mean, all these coral colonies can retract their tentacles. Sea pens are interesting. They tend to be a little bit inflated, and they can, well, perhaps retract a bit. But it looked hard. I mean, when you look at the main stem, it looks like a hard, like a rigid soft coral.
Scoop and boop.
Yeah,
Boop and scoop.
it's quite small, but we're going to see if we can capture it with a push core. No, it doesn't. Yeah, once there's little crabs in the sand too.
Well, this will be interesting. I wonder what's at the base of it.
Yeah, 9.3 degrees C at the moment, so pretty chilly. Chilly at this depth anyway, but the surrounding water temperatures are quite warm, around I think 26 degrees. We're at the northern part of Australia that's quite warm.
Is it in there?
Doesn't look like it. I see some pteropods and things. It's a little pteropod. Nope. No, we may have to.
I wonder whether it's...
Yeah, it's not obvious. Anyway, we'll keep it, of course. All these push cores are valuable. We can do quite a lot with the sediment. We can look at grain size analysis, do geochem type work. You can see it's sort of almost the anoxic layer just below that well mixed, more oxygenated surface.
It's like it's very, very cohesive mud, quite sticky.
Yeah, sticky mud, and gives that sort of classic grey look when it gets into that anoxic layer. So the other day when we opened up the push cores that we collected at the bottom of the Rodder Canyon, they were collected around 1700 metres. As soon as we split open the core, you get that almost rotten egg gas smell.
Well, that will be interesting.
Okay.
All right.
We'll see how we go.
Have a look down there.
Yeah. No, it's gone. Yeah. All right. Well, we'll head on now. You can see it's fairly bioturbated, these large mounds, but this is the shoulder, the deeper shoulder of this strange knoll-like feature. We call them the Tydeman Knoll. just because it's near Tydeman Reef. There's plenty of things dacking around. Yeah, now these sort of burrows, they remind me of the... Oh, it looks like a little flounder. There's a little flounder here, guys. You can see its eyes. You can just drop down and we'll have a closer look at it. It's just nestled in this little hole here. I was saying these burrows look like giant isopod burrows, and what we find is lots of animals make use of them after the isopod's long since gone. So we've seen some pretty big crayfish or lobsters, shrimp, fish use them. So as soon as the isopod does a burrow, it becomes a home for other animals. Yeah. So just letting the sediment waft away from us. It was fairly low current when we tested this morning. There we go. There's a good look.
Look at that.
And beautiful headshot here. So you can see his two eyes looking at us, and the tail obviously at the top. Yeah, about 10 centimetres long. And if we have a look over on the right, this is an isopod burrow, and
Anyone at home in there?
looks like we've got a crab.
Someone's in there. Crab, is it?
Crab's making use of it. Yeah, good sized crab too. Yeah, quite a hairy crab by the looks of things. Big claws. Fairly decent size. Yeah. So these animals are all making good use of these holes and burrows left by these giant isopods. Got a big sand anemone there. Yeah.
Oh, yeah.
Yeah, we've got lots of brittle stars in the seafloor too.
Is this a polychaete?
Don't know. All right, I think we should keep moving. Yep. Well, at the very start of our transect, oh, another one of those flounders, yeah, flatfish. A little bit of swinging going on. We'll just get ourselves stable here.
Kind of dust off.
So just as background, this expedition, the third expedition to the Great Barrier Reef and Coral Sea Marine Park, it's called the Northern Depths of the Great Barrier Reef. Northern being, we're at the very far northern part of the Great Barrier Reef, as far as you can go. The Great Barrier Reef stretches over 2,500 kilometres from the tip of Cape York all the way down to southern Queensland. And we are at the Cape York end of that,
screen on the map while we're just enough.
Oh yeah, okay, we got, here's a nice view. This is the 3D mapping that we've been doing over the last few days. There we go. We're zooming around. So this is actually the backscatter image, the reflectance value that we get with multi-beam sonar as well. And you can see this knoll has got quite a dark reflectance along the eastern face, and that's the face that we're going to be climbing up shortly. We're just on the ridge. You can see that white dot. That's us at the moment, and we've been mapping this area for the last 4 or 5 days now. It's pretty spectacular. Lots of canyons, underwater landslides, old river channels joining up to the heads of the canyons. And we discovered this knoll. Now that is about 1.4 kilometers in length, about 500 meters wide, and it comes up to a height of around 340 meters at the summit. So the whole face of it is about 300 meters in height. So we're going to climb across that eastern face of it and see what we can find. For us, the big mystery is, like, what is it? What is it made of? Is it reef? Is it some volcanic plug that's remnant on the edge of the Great Barrier Reef? We just don't know, and it's very unusual, so we're going to have a closer look at it.
Up until a couple of days ago, we didn't even know that this feature was there, did we? From the
No,
resolution of the previous mapping that we had. Did you want to talk a little bit about that?
Yeah, so this area of the Cape York has had no systematic mapping ever conducted before, so it's the first time that's been done. So the whole aim was to come up here and, I guess, discover what was in the deeper Great Barrier Reef. That the view shows we've got the shallow Great Barrier Reef shelf, those reds and oranges. The white behind is actually the mainland Queensland. So this is Cape York. We consider a frontier region of Australia. It's remote. It's certainly hard to get to. In the wet season, it's completely cut off. There are Aboriginal communities up here, mining camps, service towns, but it's fairly low population, and for most part it's very wild and remote. And certainly the offshore waters are the same. No systematic sampling mapping has been done in the Cape York region, the offshore Cape York region, and no ROV dives have ever been conducted in these deeper waters. So it's an opportunity to bring the Falkor up here, and this northern depths of the Great Barrier Reef expedition aims to completely turn that around, turn it from an unknown frontier environment so we can map it and understand what's living down there. Okay, lots of echinoderms there, Marty. A lot of brittle stars. We've got squat lobsters, we've seen anemones, some strange moving soft coral, whatever the hell that is. A few flounders. I'm seeing quite a few of these flatfish flounders. And we're just slowly working our way along the edge of this knoll.
Yeah. Is that a gastropod? Yeah.
Yeah, gastropod. Looks like it's underneath a bit of wood.
It's a wood. It's a wood.
Yeah, can we bump it, guys, and just see what's going on there? Because when we stop, there's a bit of sediment blows up, so we just have to wait for the sediment to be blown away from us. So we have seen in the previous dive on 396 in the Rotter Canyon, we found quite a lot of wood material that had been washed down into the lower depths of the canyon, even down around that 1700 meters. Pretty remarkable to see coconuts and mangrove roots, and we even found seagrass down at 1700 meters. So these canyons are acting like big, well, I won't say, well, rubbish chutes, you know, sediment is coming down from the shallow shelf higher up and washing down these canyons into the deep oceans. the deep ocean basin surrounding the
It's quite a different
GBR.
setting that we're in now. Here, so if it is a piece of wood or something that's settled there, is it a piece of algae? Might be algae.
Can you turn it around, please, Jara, or just have a look on the other side? So this looks like, yeah, a kind of a turban-like shell. Its foot is sticking out. You can see the operculum where it closes its lid. Can't quite make out what it's attached to.
Oh.
Oh, just fell off. No, we're not going to keep it. We're just having a look at it. It looks like a bit of algae to me. Yeah.
Tuck him in.
It looks a bit like a turban.
Let's check him.
Turban, is it turban shell? I don't know all my
I
gastropods.
don't know my shells, my gastropods really at all. When I log them in cores, I just log it as a gastropod.
Yeah, right. Yeah. Well, I like beachcombing. I only have a kind of a rough idea of what the shells are that might wash up on the ocean, up on the beach. There's some more of these flounders. There's flatfish. We've seen, I think the third one we've seen now. There's a lovely pattern on it. Big urchin in front of us. Yeah, big spiny urchin.
It's a pretty one.
Can we have a closer look at this, guys? Well, plenty of echinoderms making use of the seafloor here. We've seen sea cucumbers, there's lots of brittle stars, and now our first big urchin. And the flounder's just making a bit of a move here.
Yeah. Gee, that's fine sediment, isn't it?
Yeah, it is.
Very fine.
And there isn't a lot of current where we are. At every one of our dives we run the ADCP, which is the acoustic Doppler current profiler. It's quite a clever piece of kit, but the idea is if you keep the ship still, we fire just a thin beam of sonar sound into the water column, and it measures the change of the currents flowing underneath the hull. And that gives us our first sense of what whether the dive. Well, it's one of the first things we do when we get on station is can we put the ROV in the water? If it's over 1 knot, we usually don't put the ROV in the water. This morning it was like 0.3 of a knot.
Yeah, 0.1 to 0.3.
0.1. It was very, very low current.
It's happy days for us.
Yeah, but in fact it's quite windy still up on top. It's about 20 knots. But the current is very, very low, so that's why the sediment is kind of just hanging around us here when we put it down on the seafloor. Yep, we're getting confirmation turban shell. All right, so yep.
And there's probably about 1,000 different types of turban shells.
I'm sure there are, but you know, why I remember it is because a turban shell is one of these gastropods that have these very nice operculum. We call them cat's eyes.
Yeah, I know.
You know, when they're quite solid, and when the animal dies and the operculum's released, you end up with this beautiful white spiral on the flat side, and then it's domed on the other side.
That's right.
And people use them to put them into like carvings. Like if you go up in the South Pacific, Papua New Guinea and Solomon Islands, they often put these cats, we call them cat's eyes, but they're actually the operculum of a turban shell.
I have a pair of leather sandals with operculum decorations.
Oh, really? Yeah, okay. Well, I've spent a bit of time in the South Pacific and got quite a few souvenirs. And when you find a really, I'm always looking for the perfect one, right? One that's unbleached and has all its coloration.
So quite a lot of bioturbation here that's making these little teepee-shaped pyramids.
Now, we don't know what that is.
Better have a look at that, hey?
Yeah, so, oh, okay, so this is a bit of wood. This is a bit looks a
Looks
bit
like
mangrove.
Yeah.
Yeah, probably from the what's called the brown mangrove,
Okay.
Rhizophora stylosa.
Yeah.
So this is the very common mangrove that you find all along the Great Barrier Reef. Oh, nautilus shell in front of us, I think. Is this nautilus?
Or is it
No,
crocodile?
no, it's a
Urchin shell,
it's an
maybe?
urchin. Sorry,
Yeah.
got too excited there. So this is like a a hard urchin. They're relatively flat. They got this, they're circular. We found a nautilus on the previous dive, a dead nautilus shell, or two dead nautilus shells actually. We're in the right zone for living nautilus, right? We know that they can get up to around 300 meters depth during daytime, so they may even be on the edge of this Tydeman Knoll. So lots of these anemones.
Yeah.
Yeah, and a little fish jumping around here. Cucumber.
Cucumber.
Can we just have a closer look? So these cucumbers are interesting.
Is it decorated
Yeah,
cucumber?
they decorate their body with all of this material that's on the seafloor. So a lot of the white stuff we're seeing there, pteropods. I'm seeing scaphopods, these tusk shells. The more circular pieces are these sort of triangular shaped pteropods. Essentially planktonic gastropods. So they live higher up in the water column, in the top 10 meters or so. And when they die, they fall on the seafloor, and they'd be, when we dig down here, the seafloor, apart from mud, would have a lot of these pteropods in there. So this type of sea cucumber coats its body with this shelly material.
Well, I didn't know about decorator cucumbers. I knew that we have decorator crabs and decorator urchins.
Yeah.
I didn't know that we have decorator cucumbers. It's
Yeah.
like a crumb sausage.
Yeah. Yeah, that's right. What would you call that? Sausage in a blanket? Is that? I forget what the Aussie. And you're from New Zealand. What are they?
Well, in New Zealand, a crumbed sausage is just a crumbed sausage.
Yeah. That's,
Yeah. Our version of a hot dog, a New Zealand version of a hot dog, is a Savoy on a stick in batter.
yeah. We call them Dagwood dogs. Yeah. This is like the equivalent, sea cucumber equivalent. Pretty crunchy, I would imagine. Probably good defense, do you think? Maybe that's the reason. I mean, it looks camouflaged against the seafloor. You could hardly see it. Or, if it was an animal attacking it. We have Joan, our sea cucumber expert, with us here. She might talk about sea cucumbers later. But one of the really amazing defense mechanisms for sea cucumbers is they can spit out their intestines. They're called Cuvarian tentacles.
Oh, yes.
And if you get this
I've
stuff
seen
on
that.
your fingers,
Yeah.
it takes forever to get it off.
Yeah.
It's like the worst kind of glue, and you get it over your fingers. It's the sticky threads. They can regenerate it themselves, so it doesn't harm them, fatally. But if ever you pick up one of these shallow water sea cucumbers and it starts spitting out these white tentacles, you want to drop it quickly.
Yep.
It's nasty stuff. And of course, as a kid, we were picking up lots of things when I was growing up, and you learn very quickly to avoid these sticky threads. Snags on bread. Yeah.
Yeah.
All right, we're going to keep moving. It is pretty muddy here,
Yeah.
so as soon as we put down, we're just getting a cloud of sediment. We're continuing to move up slope. The slope being this relatively flat ridge which surrounds this Tydeman Knoll. So we're not at the knoll yet. It's still a couple of hundred meters away from us. And we've got an inset view. So again, this is the mapping that we've been doing over the last few days, or last week. The shallow coral reef is the red, orange, greens up on the GBR shelf. And what we have here is this, this very strange knoll-like feature that we're now diving on the eastern side of it. Didn't know it existed until we got here. In fact, the only bathymetry or depth data that we had prior to coming here just showed that the edge of the reef finished and then it just dropped down to around 2,000 meters. So we're absolutely amazed just how rugged and complex the seafloor is. Nice little hermit crab there. Another one of these flounders just sitting there. It'd be good to have our fish experts watching, see if they can identify this flounder. They've got really distinct markings.
So we were talking about the little pyramids in the sediment earlier, talking about biotubation. So we've got burrows that burrow down, and we've got pyramids that mount upwards, and all of those features are made by infaunal animals. Could be crustaceans, shrimp, could be worms. We're not entirely sure unless we see the animal, which we usually don't see the animal. Is that a golf ball? Was that a golf ball?
Yeah, so this is, I guess, when we're in these soft sediment environments, these markings, these life markings, they have a German name, Lebensspuren, and which just means life traces. And you know, people spend a whole scientific career studying the different tracks and trails and mounds and holes, trying to relate the animals to the tracks they leave behind. So some of them really obvious, like these really big burrows. Quite clearly, they're done by created by giant isopods, and we've seen them making them. We've seen them inhabiting them. They've got the same sort of dome-shaped burrow, and they're quite large, about 10 centimeters across. The mounds, though, are a bit of a mystery. Oh, hermit crab with an anemone on top. So this is decorated crab. Can we just get a closer look at that? Got to be careful not to put down on the seafloor. It just washes up too much sediment. That's a lovely hat.
Good morning, Will. Just saying hello to Will White on shore. He's given us the ID for that little flatfish. It's a right eye flounder.
Yep. Yeah, thanks, Will. That's great. In the family.
I'm not even going to attempt that.
Gee, that's a mouthful. Hosts a lot Saturday. Don't know if I got that one right, Will. But anyway, thank you. Thanks. We've got Will and John from CSIRO. They're in Hobart, joining us as principal investigators, our fish biologists. And we have quite a small science team on board. It's myself, Marty, Vicky, Joan, and Valerie. We had Luke joining us on the previous leg, so we only have a small science team, but we have a very large group of PIs. I think up to 15 principal investigators watching from ashore who are helping us both select samples and ID animals as we go along. We really appreciate all the help we can get here. But just going back to this biotubation, we're unsure of what might make these mounds. If I was on a really shallow reef lagoon floor and saw these pyramid-shaped mounds, I would likely attribute them to callianacid shrimps. And these are burrowing shrimps. They like living in soft sediment, seafloor, in shallow reef waters. But out here, it's a bit deeper. We're at 495 meters, so nearly 500 meters depth. We really don't know what's there. But I guess because the sediment is so stable, the the traces, the mounds, the tracks, they remain preserved on the seafloor. So it's quite an interesting environment to drift over and try and guess what's left. These Lebensspuren or life traces.
Yeah, it's interesting in modern environments like we're looking at now, modern being the current living system that we're looking at now, and it's sort of soft sediment and they're termed Lebensspuren.
Lebensspuren. Not
That's
Lebensspuren, that's love traces. As I got corrected from obviously a German speaker.
But you see in the
But we do love our life
yeah,
traces.
we love them, and we love them in the rock record too. They preserve as fossils in the rock record. They have a different name. The fossil people call them
trace fossils.
Trace fossils.
Yeah.
Trace fossils. Ichnofacies. Ichnology is the study of them in the rock record once they become fossilized.
Yeah, so the actual animal that created it is not there, but they can leave, like, let's say, mucus holes and things like that, which get infilled and eventually solidify into rock. And it is fascinating to pick up a bit of old rock geology.
And in some cases, it can be the only way of determining anything about the paleoecology, because if it's soft-bodied organisms that made these traces, they don't preserve in the fossil record. So if you were purely looking for fossils that have skeletal hard parts, there'll be nothing there.
Can we just boot it?
But those trace fossils can be the only way that we can detect
Yeah.
what was there and something about their ecology, their behaviour, what sort of groups they aggregated in, maybe if it was adults and juveniles. And so there's a whole classification scheme for these things.
That's right, yeah. It's the whole world of science around these. We're just having a bit of a look here. We saw this unusual an urchin test by the looks of things. If it is, it's very delicate. Yeah, okay, this looks like an urchin test, they call it, like the skeleton. Oh no, it's soft. It's wobbly. It's wobbly.
I'm not holding on to a word.
Yeah.
Is it hard or soft?
I don't want to crush down.
No. It looks like an urchin test to me, so...
But is it... Yeah, it does look like an urchin, doesn't it?
It has that kind of pimple look around it.
Yeah,
Yeah, it's an urchin test. Right, yes, those
Very
little
delicate.
holes would be where the spines come out. Yeah, so it looks like a dead one. We're not going to keep it,
No.
but we just saw it as a pattern in the mud.
you want to see how brittle.
Not today.
Yeah. So these are related to sand dollars. So if you think of if you go along the beach, a sand dollar is like a flattened version of these urchins, and there's a living one
with
right in
its
front of us.
spines out.
Big long spines.
Beautiful.
Yeah. So you see spine coming out of each one of these little knobs. And on top of it, you can see this little sac. Now this is actually soft. It kind of operates a bit like a hydraulic. Oh, there it goes. This is
hello.
lifting itself up. They suck water in from the top and they use hydraulics. There it is. Do you see that balloon? It's starting to contract. So that's a softer part of it, and it just it inflates and deflates like a hydraulic, like the hydraulics of machines, to push seawater around to operate all the arms and tube feet. We got a little shrimp in the background here. There's plenty. Lots of little shrimp coming in, attracted by the light. So we're 500 meters. It's pretty dark down here. We're definitely not in the photic zone or even the mesophotic zone. And it's 9.8 degrees C.
Oh, hello.
Oh, what do we got here? Oh, gee.
Wow, that's quick.
Yeah.
Up and away. A game of tag.
There's a lot in the water column
Yeah,
here. Look
there's
at all
a
this
lot of
shrimp.
It was a fish. Is a fish. It is quick.
Yeah.
I think he's away.
Yep. Only got a glimpse of that.
Yeah.
It's an eel of some kind.
Yeah. Is that him there?
Oh, is that Anthomastus? There we go. There's our eel.
Is it? Is it a flatworm?
A flatworm.
Yeah.
Pretty long one if it was.
John says Synaphobrachidae.
Synaphobrachidae.
Basket
Yeah,
wheel
basket
eel.
work eel. Yeah. Thanks, John. Yeah, and we have seen, so basket work eels or cutthroat eels, same family, I believe. And we have seen a lot of those in the deep Great Barrier Reef and Coral Sea in all of these voyages. Seem to be common feature of the fish life in these deeper depths. Here's one of these disappearing act.
Ah, yes. Now, Jeremy, now that you're sitting in your office at your desk, watch this.
Yeah. All right. We might see if we can. This we've tried twice now to grab one of these, but it disappears into the sand. We do not know what it is. It looks like it's got soft coral around the outside. There it goes.
There it goes.
The heck
Retracted.
is that?
So there has been a couple of suggestions that it's a sea pen, but it looks like that central stem is sort of rigid, doesn't it?
Yeah, it does. Yeah,
I guess
it's not.
Can a sea pen have a rigid stem? I guess.
Well, it can. I think they can inflate. You know, they
Oh yeah. With
they have water to help
that
inflate.
hydrostatic pressure.
Yeah.
Again.
Yeah, with some say sea pen.
Yeah.
Okay, interesting. I tend to think of sea pens as a sort of big feather-like,
Feathery,
you know, with quite broad, but that's very narrow with the polyps just along the main stem. Okay, we'll go with sea pen.
Yeah.
That's good. Yeah, thanks to everyone ashore helping us ID things here. So I'm a marine geologist. I'm more familiar with rocks. And Marty, you're a jack of all trades.
Oh yeah, a bit of this and a bit of that.
Yeah.
Carbonate sediments for the most part, but carbonate sediments are produced by biota. So
So you have
there's
to
a bit
be
of
familiar.
crossover there.
Yeah. There we go. There's another hermit crab with an anemone on its back. A fascinator, I think they call them.
Oh, yeah.
At the races.
At the races, yeah, your
Yeah.
frilly little feather hat at a jaunty angle.
Yes. We've seen a few, like the other day we had one with a real Mexican hat style. Sombrero anemone.
Another little crab.
Oh, okay. This is an interesting crab. It's kind of dome shaped. It's tucked its claws in. It's like no one can touch, no one can mess with me.
He thinks that we can't see him if he does that.
Yeah, I bet it's got a decent set of claws on it too.
Alright, we'll continue on.
Yep. So you can see every time we sit down, it becomes quite. The sediment really jumps up in the water column, so we'll just have to be careful about how we do that. But we're hoping that once we get to the edge of this this large knoll feature, that perhaps the sediment will disappear. Because
Yeah,
one
it'd
of
be
the
interesting to see if the slope is such that the sediment can't settle on it. Although we've been
Yeah.
surprised on the canyon walls, haven't we? Even the steep slopes have still had that fine sediment covering.
yeah, it's amazing how steep it can be, and yet still have mud,
Yeah.
thick mud on the sides. But we do like steep and deep because steep tends to over, where you get you get it oversteepens to the point where sediment can't build up, and then you get these exposed rock faces, and often find lots of marine life, sessile marine life attached to it.
Yeah, in geology, sedimentology, it's called the angle of repose, and when everything is settling under gravity, so the angle becomes too great for the force of gravity acting upon the grains, then they can't settle, and they'll tumble down under gravity. But then, of course, mud is so cohesive and sticky, even though it's finer grained, it can overcome that, or its angle of repose can be steeper.
But at times when the SuBastian actually touched down in places, it doesn't take much of a push to get it going,
moving.
And it just releases. And at the base of that, in the axis of that canyon that we were the other day, you could see big clumps of mud that have dropped down into the axis floor.
Yeah, and they were sizable chunks, and they'd even started to get that ferromanganese crust over them. So they had the appearance of rocks. We were thinking, oh great, this is a hard substrate. There was still things attached to it, but when we gave it a poke, it was really soft.
Very soft, yeah, but it was sort of cohesive soft, like the strata had just given away and dropped down as a big block onto the lower seafloor.
Keep going.
Yeah, so we're just traversing a lot along the lower shoulder that surrounds this knoll-like feature. So relatively flat compared to where we're headed, and the idea was that we do this traverse so that we can get a good sense of what was around the base, and that way it gives us a better understanding of, I guess, the change in habitat once we are on the knoll itself. So we expect that it'll become a lot steeper, but we don't know what it's made of, since that's one of the goals of today's dive, is to try and understand what this knoll is made of.
And if it's covered in mud, I think we should scrape it away and try and see what's underneath if we can.
Another one of these
crab, decorated
crab? A crab, but with one of those really interesting gastropod snail shells. It's like the whorl part, the internal part of the gastropod's the only thing remaining. It's like the outside is completely fallen off.
That's a question. Would Croxus be this far out from land? That is an excellent question.
Well, the answer is yes.
Yes.
It was a guide chewed at Lizard Island only a few weeks ago. Now, we passed Lizard Island on the way up, and Lizard Island sits on the outer shelf of the Great Barrier Reef, and in fact we're close to Princess Charlotte Bay, which is a big estuarine environment. Yes, so the answer is yes. Crocodiles may not be out here swimming in the deep water, but certainly they are traversing up and down
long distances, don't
the Great
they?
Barrier Reef, looking for new homes, areas for mating and things like that. But they
been studied with radio tracking.
They do travel long distances. Of course, they spend most of their time in saltwater estuaries close to the coast. But I guess the point being that yes, they can migrate up and down this zone. And if we're about 70 kilometres away from the coastline itself, and if we were to be on that coastline, there would be many creeks and rivers that have got crocodiles in them.
Yeah, as a scuba diver in the tropics, I'm not worried about the sharks really. It's the thought of a looming crocodile that terrifies me. Oh, here we go. Look at this.
Yes. Well,
this is what we're looking for.
Yes, this is interesting.
So we are right at the base now of what we're calling the knoll.
Okay, this is.
And here we are.
Looking at it. Okay,
right.
Dark is this ferromanganese oxide.
Yep. Next question is it hard?
It looks hard.
It looks like it's
Can we give this a poke, please, J-Rod, so we can just see what we're right at the very base of this knoll.
Yeah. They look like veins. Joints with veins, if they're hard.
So we're going to be on this rock now for most of the day. It's got a thin covering of sediment over the top. See, it goes up pretty steep from here.
Got these parallel lines to me, pending whether this is soft or hard. They look like joints that have been infilled. Joints and veins.
So just going back to the crocodile, just to finish that question, crocs are air breathing, so they need to be near the surface to be able to breathe and survive. They would not be down this deep at all.
Oh yes.
Hard.
Fantastic.
Okay.
Okay.
Well, that's good to know.
Well, we definitely want to try and sample this, so sorry for the folks watching at home, but there might be a bit of sediment blown around here for a little bit, but if we can find a spot where...
If we can put down hard rock, Marty. So this is very different to the sort of thing we're looking at the canyon the other day.
Very, very different, and this doesn't look... I mean, the other day we were looking at turbidites, which are layers of sediment, soft sediment, but this is very different.
Kind of just broke a bit there.
Yeah. Is it coming off? Now, if that is a joint that's been infilled, it might be filled in by a different material. Oh, there you go.
There we go. That really is a bit of resistance there.
Yeah. All right. Well, we'll keep it.
Not much. Not much.
Yeah. Yeah. We'll try and get more.
Try and get a bigger piece.
We got the rock drill too.
Yeah.
Just looking for somewhere suitable to be able to get a grip of what's on this rock, you know. So we're looking for joints or cracks. Oh, is that a... It's not a coconut, is it, Marty?
Is it a coconut sitting there on the bench?
Just a fine covering of silt over some of this stuff. We saw coconuts at the bottom of the Rotter Canyon at 1700 meters the other day. So vegetation gets washed out off the Great Barrier Reef shelf and then eventually becomes waterlogged and drifts down into the deeper waters. Yeah, so this knoll feature, we're calling it the Tydeman Knoll because it's close to Tydeman Reef. It's about 6 kilometres away from where we are now. It kind of pokes up out of this big broad ridge. The ridge itself drops down to 1800 metres, so nearly 2 kilometres deep either side of us. And the knoll itself is about 1400 metres long, so nearly 1.5 kilometres long. It's about 0.5 kilometre wide, and it rises from this depth around 460 metres up to 340 metres in the kind of lower mesophotic zone. So big question is, what is it made of? And so getting a rock sample is pretty important. We can do a lot with those rock samples. We can, if it's organic material, we can do radiocarbon age dating on it. We can do all kinds of geochem work on it to to understand its composition. Like, is it biogenic, meaning is it limestone, or is it something even more weird, like, you know, volcanic origin or even granite? But that, I think, is unlikely because the deep geology of deep geological history of this area doesn't lend itself to igneous intrusions or volcanic eruptions on this part of the Great Barrier Reef. So that would be quite unlikely. But it's clearly, it's very old looking. We're not seeing a lot of layering, which is what you'd kind of expect if it were, say, a drowned reef, an old limestone edifice, you know. But the dark colouring is ferromanganese oxide. This is what you get on rock that's been exposed to the sea for a very long time. These little stylasterid hydrocorals. Yeah, yeah. These little, delicate little corals, they're stylasterids. They're hydrozoan corals, and typically found in these water, the depths. Okay, that's a better rock. Okay, we want to just have a look at the broken off bit. Okay, that's pretty good. Pretty good. Any thoughts, Marty? Looks bioturbated, you know, like, is it burrowed, bored? Could it be limestone, do you think?
Could it be limestone? It could. I mean,
if it's limestone, we're talking about a drowned reef.
Oxidated.
Like
it's been oxidated.
Yeah.
Orangey mustard colouring.
So when you do geology, it's important that you try and get as fresh a face as possible, something that's away from the outside edge, which can confuse any geochem work or age dating. So we'll definitely use that.
That broke off relatively easily.
Yeah.
1A, please.
And we've got lots of tools up our sleeve. I think of the ROV SuBastian like the Swiss Army knife of ROVs. We've got a rock drill, we've got brushes, we've got little scoop nets, paintbrush type implements, anything. We can pull these out and use them to help us understand what's on the seafloor, collect things. So we may pull out a rock drill at some point during the day.
We all
much.
want the rock drill to put in an appearance.
Yeah, yeah, it worked very successfully. In fact, on the last expedition with Geoscience Australia, it was pretty thrilling. We've got to a bit of outcrop rock that sticks out at the very base of the Great Barrier Reef.
Yeah,
so
continental
rock.
Yeah.
And we drilled a piece out of it. So it's probably the oldest bit of, well, the base of the Great Barrier Reef, you know, where from which the Great Barrier Reef is built up upon.
What is that?
Little scorpion fish, perhaps? Squat lobster. Bit of a sponge up here on the right.
Yeah.
But we're seeing these stylasterid corals starting up here.
Yep. We'll just carry on.
Yeah.
So, okay, if this potentially is a drowned reef, we need to keep our eyes peeled for other possible layers that we might want to sample from on the way up. Yeah,
the layering is a bit of a giveaway. In fact, the transect we chose, Marty, if you remember on that, as we get along this eastern face, it looked almost like a scarp, a landslide scarp, where a bit had broken away. So that'll be interesting to have a look at.
What these are.
You know, they look kind of sponge-like. I wonder if they're these rock sponges.
Oh.
That looks like that too.
Is there?
Yeah. Xenobite.
Yeah, it looks like it here.
Yeah.
Okay.
So I like these vertical faces where it's too steep for the sediment.
Yeah.
There's lots of things
Check that out.
in there. We've got Chris and J-Rod on the ROV controls, helped by James on the wireless.
So we're keeping on these hard surfaces, we're looking out for sponges,
Yeah, and any
hard corals, black
and soft
corals, our usual list. Isn't
Yep.
it?
Yep.
Oh,
Some
here we go.
Oh, no, it's like a soft coral.
Let's go check it out.
Yeah. And spider crab sitting behind it.
Yes, please.
Now, I'm not a soft coral expert. Is it Chironeptha? Is that
haven't-
the
no.
soft coral?
I have no idea.
They actually did a deep sea thing to a park one time.
Did you?
Or is it a sponge?
Or is it a sponge?
No, it looks octocoral like to me, soft coral like. You can see the polyps on the side.
Can we like
I think we want to.
see whether there's like tiny critters on it?
Yep.
If it has a shrimp on it, we're thinking about it's not freezing.
Okay.
Is this a genome sample?
If it has a shrimp on it, but otherwise, probably not.
Oh, that's just a view.
Yeah, so our sampling is, we have our standard sampling where animals go into bio boxes and they're preserved in ethanol. But then we're working with the Sanger Foundation and Smithsonian Institute to collect samples that will be flash frozen in liquid nitrogen. That way preserves the DNA much better than you can with ethanol, and they have to be collected separately and stored in a special quiver that we've got on the side of the ROV. Yeah, we're looking at getting up to 340 metres. That's the summit of this knoll, and no higher. So from the mapping we did last night, the knoll depth is around, well, 450, 460 metres depth. The top of it's about 340 metres. So we're going to do this long horizontal transect for most of the day, and then we're going to reach the crest of this knoll, and then we're going to work our way up to the summit. So we're definitely mountain climbing today. And we're just having a closer look to see if this is one of these these corals that we want to sample for the DNA project.
Jeremy is standing by. He said no need. He said no need.
Okay. No need. We'll just keep moving. Yep. So we've got Jeremy Horowitz, our other veteran Falkor participant. He couldn't make it on board for this leg, but he's watching from on shore. Yep, tell him that we'll go anytime. Yeah, got a lovely big spider crab coming up in view now. And we got lots of people watching, so we're going to try and get Jeremy Horowitz joining us on audio from his home in Townsville. Wow, look at the top of this, Marty. Yeah, okay. So what we've got is a face which is broken away, and the top of it has a really distinct ferromanganese oxide crust, which suggests very old. You know, it's been exposed to the seawater here for quite a long time, and you end up with this classic crust over the top of.
One of these.
Yeah, this looks like a rock sponge to me. This sort of ear shape. I think we've seen them before. But this crust just looks, it looks like ferromanganese oxide covering to me.
Yes, please.
We'll see if we can break it off.
Do you want the crust?
We can do both. Yeah.
Definitely what it's on, not away with it as well.
So we're just going to come and see if we can get a piece of rock here. At the same time, we're trying to organise to get Jeremy Horowitz to join us in audio from the shore. We can go on headset.
Yeah.
Oh, pretty hard though, was it, Chris? Like you could feel it. There's a bit of resistance there.
He was using the arm, so I'm not really sure.
Oh yeah, J-rod's on it. Sorry.
Should be able to get it. Stay around with it. Good. Yeah. That's locked in. I was gonna try to fly if you need me to.
So J-rod's on the arm. It's pretty hard, isn't it?
I'm not gonna take these two arms, but you don't want a piece of the bottom stuff as well. We will put
But it was in one beak.
It's very dark, isn't it? A really distinct crust.
Where do you want this one to go?
One beak.
Pick up all those little fragments over there.
Yeah.
You gotta reach where you need to find out.
Some big cup corals up there too.
Someone will may be interested in that. If not Jeremy, then it's one for you, Sco.
You don't want a couple?
Yeah, we do.
Put this one away, maybe just start around. That's, I'm not gonna have to cut it.
to me, in which case I can just go in with those guys. So unless anyone else shouts out that they want it.
It is a dead
Oh,
one
is it
though.
dead?
Yeah. I think Yusuke wants a live one.
Yeah.
Yeah.
No, okay, leave it.
No, we'll skip that one. Thank you. Just get these last fragments, put them in our bio box. We'll be at the end of the dive each day. We swarm around the ROV and bring all the samples into the wet lab here, and then it can take a few hours of sorting and preserving and all the recording that's required to keep track of each individual sample, because they have to go to different parts of Australia and around the world. All the different taxonomic experts, geological experts who will be working on the different samples that we collect. Yep, thanks, Joe. Almost at our start of our second leg here, in the big trees. Yeah, so we're now at our. We've planned this very long transect along the steep eastern face of this knoll. So some things we want to look for. We want to look for stratigraphy or exposed strata. If this is a limestone, or like a drowned reef, then we'll see evidence of layering. So far I'm leaning towards a drowned reef. Yeah, but quite an old one, given by the amount of ferromanganese oxide crust over the top of this rock. Alright, so we're just getting ourselves organised here, and then we're going to lift off, and hopefully we'll have Jeremy joining us pretty shortly. Yep.
Hi there, Jeremy. We've got you joining in now via Zoom.
Hey, John. Hey, Rob, Marty, everyone. How's it going? Can you guys hear me?
Hey, Jeremy. Can you hear us okay, Jeremy?
Yep.
Okay.
I can hear you fine. All right. Let's see what we got here.
Wow, yeah, look at all that in the water. It's like a little stylus thread under that underhang there. that big urchin.
Jeremy, can you hear me?
Yep, I can hear you. Nice. I wonder if we could zoom in just underneath this crest, like just underneath the face. Maybe you could look for little small conifer sponges, little things maybe hiding.
Folks that are watching the live stream, we're just doing a little audio change around here, so bear with us for a moment. Different audio setup when we have our onshore scientists joining us via Zoom.
All right, Jeremy, just testing again. Testing 1, 2, 3, 3, 2, 1. Can you hear me?
Yep. Hey, Rob, I can hear you. I can hear you fine.
And I can hear you too.
How's everything on the ship?
Yeah, everything's good here. We're all just putting on head mics so we can talk with you, but not over the top of you. So we'll turn off. We'll just put it to standby as you're talking, and we can jump in as is needed. So we can hear you and see you fine.
Fantastic. Yeah, okay. It seems like it's pretty muddy. So I imagine it's a bit hard to get low to the ground to look outwards. Is that correct?
Well, it's actually just a thin veneer of sediment. Yeah, it is fine sediment. What we found as we sit down, the current down here is quite low. We're on the protected eastern side of this knoll, and we found that as it sits down, it kind of washes up some of the sediment, but it's actually not very thick at all.
Interesting. You okay?
So we're just trying to maneuver the ROV now to get a closer look underneath this overhang.
Yeah. Yeah, looks like a good area for things to potentially be missed if not looking really close.
Is that a sponge sticking out on the edge of that knob, sort of center left of screen at the moment?
Center left. Potentially, maybe zoom in to that.
Just give us 1 sec. We've just spotted a very nicely accessible bit of rock sticking out. So we're going to go for that, and then we'll go look at that little potential sponge question mark maybe.
Yeah, okay. Well, the interesting thing about, the good thing about collecting these rocks is that almost every rock that we collect, even when we look at it in the water, when we get it on the ship, there's always little things living on it that we just can't see even with these cameras. So I'm sure Joan and Valerie will have the chance to look at the rocks when they come up before they're scrubbed.
Promise not to start scrubbing until the girls have come and had a look.
Yeah. Well, we've had a lot of interesting things on the last trip on the rocks.
Especially one of the really small little sponges was on one of the rocks, wasn't it? One of those tiny little coniferous sponge.
Yeah, and that's what I'm looking for now. So Merrick Eckins at the Queensland Museum was so excited to see them in person because, coniferous sponges are collected from the deep sea, but usually they're collected from dredging. And these things are fragile. They crumble under your thumb. So in our ability to grab them and put them carefully in the bio box means we have technically the first, perfectly preserved samples that can be used to describe these undescribed species. So it's a great opportunity to, look close at all the rocks and underhangs and on the rocks that are collected for other reasons. Every opportunity to, spot these little guys.
And the great thing, of course, about collecting with the ROV is that you get the situational context of where the sample came from, and everything that it was surrounded by, the community, as opposed to when you bring up a dredge sample and things are smashed around a bit and you really aren't sure what was with what.
Exactly. And that's why this trip is such a perfect opportunity to also sequence genomes of species that live together, either within each other or on top of one another, because we know what we're collecting, and if we put each pair of individuals that live together in an individual bio box or in the coral quiver, we can be confident that they do live together, and therefore we could sequence them and try to understand how they have co-evolved, which is the main purpose of this project that we are lucky to be a part of.
So, Jeremy, do you want to talk a little bit more about this genome project? You're quite involved with it. I mean, we're doing all the sampling and preservation, but it'd be really helpful if you can explain it a bit more, please.
Yeah, sure. Actually, John, do you mind putting up, if it's possible, some of the images I sent you earlier? So this project we're doing, it's a collaborative project. It's called the Aquatic Symbiosis Genomics Project, and it's run by the Sanger Institute, and they support genomic research. And so the goal of this project is to read the genomes of thousands of species that have some sort of symbiotic relationship with other species. So our question that we're trying to answer is, how do these species evolve together? And so symbiosis can either be a mutually beneficial thing like, coral and algae, or it can be parasitic. So mosquitoes and humans are a good example of, a parasitic interaction. And so, evolution is the fight for survival, but some species have seemed to kind of team up to survive together. So you can see pictures here of corals that have either animals living within them, or they have the symbiotic algae that they're living together and helping each other. So if you could pause on one of those branching black coral pictures, the one with the branches coming around, that one, yep. So if you could see that one, yeah. So you could see in that picture, so this is a black coral that has what we call worm runs on the stem. So you can see, if that picture is larger, you could see that the branches around the stem have been bent by a worm. And so the top right image, you can see that there's a thin stem and then this circular outcrop around it. And those are branches that have been folded carefully by this worm. And so this worm lives within this species of coral. So it's really amazing. And we never find this coral without a worm run, which is amazing, right? There's obviously lots of worms, and I guess whenever they have the opportunity to live within this coral, they do. So there's probably a really interesting story about how these two, a coral and a worm, have evolved over time to have this mutually, or either it's mutually beneficial or it's beneficial to one and not the other. But to understand that story, how did that come to be? How are these species evolving? So we can compare the relationships between closely related species between these distantly related animals. So these are the kinds of corals that we're targeting. Yesterday we collected soft corals and sponges that had some sort of associated animals with them. And we're going to be looking for more of the same. So this picture, the one that's pictured here, is a really interesting target. We saw it a lot on the last expedition. And now we have liquid nitrogen. So we have the opportunity to sequence the genome of the worm and the coral. So it's really exciting. So it looks like we're collecting a rock here. Seems like a hard rock.
Yeah, we think that this is a very old, ancient reef limestone. It'll be cemented together with calcite cement, which makes it hard. So we're just going to have one more crack at this and then leave it be. We'll get on to zooming in on all those potential targets hiding in amongst these little cryptic cavities and underhangs.
Yeah, and so besides collecting for this exciting genomic project, we're also doing what we did on the last trip, which is simply collecting deep sea species to help us improve our understanding of the taxonomy and the evolutionary history of these various anthozoan groups. So the sponges, the soft corals, the black corals, and the hard corals. And even yesterday, we came across a coral that we didn't see on the first expedition. So obviously, we've been collecting a lot of the common species, but the more dives we do, the more chances we're going to get of finding rare species that do occur here, which will help us understand the range expansions of the various species that live in the coral sea. And that information is what we use to directly update the marine reserves and the strategies that are used to preserve biodiversity. Okay, looks like they're giving up on the rock. The rock was too hard.
Too hard, literally. Now, Jeremy, you want us to just pause here and look in and around this area, right? We're particularly looking at the small stuff and the nooks and crannies and under the overhangs.
Yeah, maybe just like a really quick scan while you're zoomed in, just underneath that flat top overhang. And Google Lindsay Medical are going to be sequenced for their full genomes. We're flash freezing them. So essentially, we're dropping the temperature down below -80 instantly, thereby preserving the DNA, which would otherwise be degraded. And they'll go in a dewar, which is a container that contains liquid nitrogen, and that tissue will just be preserved until we get it back in the lab, and then we can extract DNA from that and get the highest quality and quantity DNA possible. It was quite a a task to get the liquid nitrogen on such short notice, but we did it. Thanks to Joan and Valerie helping us out. So we now have it on board, and it's just a fantastic opportunity to preserve the tissue of these deep sea specimens that really generally, it's rare to have deep sea specimens that give us good DNA for molecular work. So we're going to learn a whole lot about all these different species that to date isn't understood.
So yes, it's quite beautiful under here. This pink coral in front of us with the white tips, it's a stylastrid hydrocoral. And the delicate white corals around it also stylastrids.
Yeah. How about that netted, maybe that's a bryozoan just to the left of the stylastrid? Maybe it's a bryozoan?
Yeah, possibly. Just above that crab claw.
Yeah.
Lots of sponges in amongst the nooks and crannies.
Yeah. Yep, nothing carnivorous looking. Okay.
Okay, should we zoom out and keep going?
Yeah, sure. Let's do it. I see people commenting that they're interested in a career change. There's definitely a lot of work to be done in these fields, these various fields that, you know, help better understand deep sea. Especially the technology that we have now, where even 5 years ago we couldn't answer questions that we can now answer fairly with, you know, without too much effort. We now have molecular tools that give us really high confidence in our results, when before, you know, we would have these what we call phylogenetic trees, or depictions of the relationships between species, but really low confidence factors on those trees. So yeah, now we're able to really start answering questions, these long-standing evolutionary questions, like how have these different species evolved, you know, and what do they relate to? Really large, fundamental questions that over the next 5-10 years, we're going to answer.
Yeah, that's right. I mean, talking about fundamental questions, for me, it's always about what is the shape of the seafloor. And so you have to bring in this modern mapping tools like the multi-beam sonar on the Falkor to just systematically map the seafloor and get a sense of what's down here. So Cape York, this whole the whole focus of this expedition has never been systematically before, and so it's a chance to get up here and see what it looks like. And we've been really astounded at how rugged it is, just huge canyons. This knoll, well, we're thinking it's a drowned reef now. Didn't even know it existed until a few days ago, and now we're actually diving on it. So it's pretty amazing to go into, you know, I guess a new part, an unexplored part of the Great Barrier Reef, and just see what's down here.
Yeah, absolutely. And I know you've been mapping the Great Barrier Reef and the Coral Sea for a large part of your life. What percentage of these deep sea habitats have been mapped at this stage? And I guess how much more do we have to go to map it all?
Yeah, good question. I mean, the Great Barrier Reef Marine Park area itself is around 350,000 square kilometres, so it's a lot of area to map. It's not just the shallow coral reefs, of course. The marine park extends out into these deeper waters. So the shallow shelf area is actually around about 40% mapped. Most of that has been done by LiDAR, airborne LiDAR, conducted by the Royal Australian Navy and the Australian Hydrographic Office. So most of the shallow coral reefs are quite well mapped. These deeper waters, though, in some areas like Cape York, where we are now, very poorly mapped. Still lots of canyons yet to discover, but for myself and my colleague Jodie Webster at University of Sydney, we've been systematically mapping the margin, the continental slope, the deeper waters of the Great Barrier Reef since around 2007 with modern multi-beam sonar. And some parts of that margin are very well mapped, like from Cairns up to Cooktown, stretch of around 100 to 200 kilometres long, quite well mapped. But north of around Cooktown, it was all unknown. So it's really exciting to be here in the Falkor and do this mapping to find out what the shape of the seafloor looks like. It's very eroded, of canyons, deep canyons, down to around 2,000 meters deep. And then you have these surprises like this kind of appears out of nowhere.
All right, no, it's really exciting.
So Jeremy, this just to give you a sense of the physiography here, the knoll itself is about 1.4 kilometres long, about 500 metres wide. Base of it is at 460 metres, and the crest of it is at 340 metres. So it's it's 200 metres high. We are traversing along the eastern face of it, so it's steep. This is where we're going to be most of the day, and our aim is to head around to the crest, and then we will climb up to the summit. So it's very much like mountain climbing, today, and we're just looking for any soft corals, hexacoral, anthozoans that we can sample along the way. Looking for overhangs these animals like to attach to.
Yeah, I guess we don't really know what to expect with in terms of biology as we get to the top of this knoll. It'll be exciting to see. Is that a cup coral right there on the?
Yeah, it looks like cup coral. Is that one of your targets there, Jeremy?
No, not necessarily. There is one target in particular that on the last expedition we found this black coral that occurs really mainly just in the sand, of course attached to a little rock that's kind of buried below the sand. And after looking at it in the museum, it's very clear that it's very different than we thought it was in terms of its family relation and even its genus relation. So it's a species called Platycalicus, and it could occur from 400 meters to 200 meters. The challenge is it's so thinly branched and wispy that it's really, really hard to see, which is why one of the reasons why we probably don't know much about it. But it has these really unique features that no other black corals have. Notably, on the base of the colony, it has these long, long spikes. They're maybe a centimeter tall, and they cover the base of the colony, and no one's ever had a good enough specimen to observe these spikes. But we collected 2 on the last expedition, and I have since extracted DNA from both of those. So we'll soon sequence the DNA from those 2 specimens, but it'd be great to get a few more just to see if we're looking at one species or multiple species that look very similar, and also try to pin down, where it falls in the order of black corals, which family it belongs to. So I'll keep an eye out for that one as well.
Righto, Jeremy. Yeah, we're just looking at some quite strange seafloor. It looks like an infilled fault of some kind. It looks organic, crusty material in the split in the rock.
Mm-hmm.
And it's vertical.
Yeah, all this organic material that's kind of found its way to this area. I guess it could be limiting the number of coral species that live on this knoll, at least on this side.
Yeah, that's right. I mean, there's just a thin veneer of sediment here, but that might be enough to stop corals settling. Certainly these steeper faces are conducive to sessile animals attaching to the rock. But yeah, there's this thin coating of dust everywhere. It was quite muddy down at the base of this knoll. Just going to pull ourselves away from the wall a bit, get away from this this sediment that's been washed up.
Right. Rob, you could probably answer this question about this part of the GBR. Is this area a part of the GBR marine park, or is it too inaccessible for people?
Yeah, it is part of the Great Barrier Reef Marine Park. So the the marine park extends out into these deeper waters, but it is very inaccessible. So most people visiting the marine park travel along the shelf, on the shallower shelf in between the shallow coral reefs. Very few people get out into these deeper waters. So we've been operating out here for a week now, and we've not seen any other shipping traffic in these deeper waters. We've seen a few ships pass us on the shelf itself. It's called the inner route of Great Barrier Reef, and it's a compulsory pilotage area. There is some merchant vessel traffic travelling through here, but we've not seen any other vessels. Certainly the like adventure dive vessels occasionally get up into these waters a couple of times a year. There's also Rain Island, which is at the very tip of Cape York. We'll be heading up that way later on in the voyage. So Rain Island with the Galapagos of the Great Barrier Reef. It's one of the largest green turtle rookeries. So we'll be up and around that region. So certainly people like national park rangers go up to these very isolated sand cays to do their work. Out in these deeper offshore waters, very few people get out here. From a mapping perspective, only about 4 or 5 multi-beam have ever been through this region. Hence, it's very poorly mapped, and that's the reason why the Falkor's here for the next 2 or 3 weeks to map this quite remote area. We are calling it the frontier of the Great Barrier Reef for that reason.
Right. Well, it'll be interesting when we get close to Rain Island. Maybe we'll see some turtles during the ROV dives.
Yeah, possibly. You remember they are shallow. They're all air-breathing reptiles, of course, so they never swim down real deep. We do occasionally get the SuBastian up into these shallow waters, the mesophotic zone. I think we've got SuBastian up to around 40, 50 meters in places. Typically, we like SuBastian down deep where it's cooler. We're at 11.9 degrees Celsius at 417 meters down. Seems to be a nice temperature for the ROV SuBastian to work in. And I talk about that because in those shallow waters, it has to work harder to keep it stable in the currents, and it can easily overheat because there's so much machinery and hydraulics and electrics inside SuBastian. You don't want to spend too much time in those shallower waters.
Right. Maybe we could do a zoom in on just any face of this side here. Just a quick zoom in to see what is covering these rocks.
Yeah, I'm seeing cup corals, both alive and dead. Look like some oysters, perhaps rock sponges. Seeing dead cup corals on the ground. These look like rock sponges to me, these sort of large, lumpy masses.
Yeah, like encrusting sponges. Yeah, okay.
And these delicate white colonies are the stellastrid hydrocorals. We found them typically found them in these depths in other dives, so we're quite familiar with what they look like.
Mm-hmm. Yep. The ones we collected on the last trip are now at the museum, happily in ethanol.
Valerie and Joan, we had our first go at it yesterday. It seemed to go quite well.
Yeah, I'm glad that worked out. Yeah, actually, I've never worked on liquid nitrogen myself. It was a good learning experience for me, learning how to use it and then showing them how I think it can be, but I know you've used liquid nitrogen before, so I know they're in good hands.
Yeah, we've got plenty of good safety protocols here, and we've got Vicky, who's also an expert in the laboratory, sort of keeping us all in line. That was good fun.
Very good. Well, someone in the lab needs to be watching everyone else. Of course, that was me on the last trip, making sure everyone was safe.
Mr. Workplace Health and Safety, that's you, Jeremy.
Figured you would laugh at that.
Yeah, got a good question here about the water temperature. As we have moved north, yes, the water temperatures at the surface have slightly warmed compared to down south. I think we're up around, what, 26 degrees-ish? Marty? Yeah, at the surface.
Water temperature at the surface? Yeah, that sounds about right.
Yeah, but as you get down, as you get into these depths, it becomes a lot more stable, right? So then we're dealing with different water masses below those surface water masses. So right now, 400 meters, we're probably within a subtropical mode water. Would that be right?
Oh, that would be my guess, just by the depth. I mean, we're not looking at any salinity or what have you here, but that would be my guess.
Yeah, and because we're dealing with depths of around 2,000 metres, not far from where we are here, we go through two other water masses before you even get to the seafloor. So we go from surface warm waters, which is, you know, what you'd swim in, of course, and then we get into the subtropical mode water, and then around about that 400 metre mark to around about sort of 600 metres, we get subantarctic mode water, and then Antarctic intermediate water at the bottom depths. So it's really cold down there.
Okay, we could maybe move up a little bit.
Got ourselves look like a reef fish sitting in a hole here.
Oh,
kind of had it looked like a bit of a blenny or goby type head. Anyway, hoping that the fish people are paying attention.
I think someone in the fish army is always paying attention on these expeditions, which is great. Who is that?
Yeah, we've got John Pognoski and Will White from CSIRO coming in. Fitiform. Folks are little neobithites. Like, yeah, there's a bit of a glimpse, unfortunately. Will, it kind of ducked into the hole, but we'll keep an eye out for any fish in these nooks and crannies as we go along. So this face is very eroded, which we're leaning towards old limestone substrate here. So we are thinking this Tydeman Knoll is an old drowned reef, which is fascinating to think that it existed here for all these millions of years, and no one knew it was here until we came along and mapped it. So this is a fairly large drowned reef. Drowned in the sense that for whatever reason, it wasn't able to keep pace with any changes in sea level, and is now drowned. The summit of it is at around 340 metres. We aim to get to the top of the summit today. This reef, it would have existed as a shallow coral reef in the long geological past, but here it is now just preserved on the seafloor and fairly eroded. You know, the animals, polychaetes, worms, mollusks just boring into the outer surface and leaving this very pockmarked surface. Oh, we're going to keep moving along. We're just following this long kind of horizontal transect along the eastern face of the knoll. Yeah, here we have the inset. So this is looking down on it from shore, from, you know, if you were standing on the Great Barrier Reef itself, this is what's in front of you. So it's kind of knife-like. It's about 200 meters in vertical height. Either side of it, though, drops right down. Those canyons either side of it are nearly 2,000 meters deep. So this is the area that we've been mapping over the last week. That's this sort of grey-blue colour. And the red-orangey colour is the mapping that was done by the Australian Hydrographic Office using LiDAR. So that's the shallow shelf region. So those red reefs you could scuba dive on. This very large knoll feature that's about 6 kilometres from the edge of the Great Barrier Reef. We are currently diving along the eastern face of it at the moment. And we're just zooming in now. See how rugged it is. Yeah, that's right. Who knows? Is it just sand, mud, or will there be lots of soft corals and the like? We don't know. But,
could it be like could it be like a pinnacle? You know, where you have lots of high diversity at the tip.
Yeah, we're hoping so, Jeremy. I mean, it could act a bit like a seamount-like feature. These conical features on the deep ocean floor, and they, affected by whatever currents are sweeping past and eddying behind them, often bringing in lots of marine life, taking advantage of the currents that they interrupt. So, is that what this knoll is doing? Well, by the end of the dive, we hope to have a clearer picture of what's living there. Oh, pycnogonid.
I think
pycnogonid here on the right. So this is a sea spider. It's not a crustacean. It's distantly related to arachnids, I believe, although maybe some experts can tell us a bit more about it. That's pretty big, it's a good 10 centimeters across. We're going to have a closer look at it. A classic sea spider.
Question on the YouTube chat feed there about who named the knoll, since it was only just discovered. Well, we named the knoll on the fly the other night as we were looking at the mapping, and Rob named it, at the moment we're calling it Tydeman Knoll, seeing as how it's directly adjacent or in front of Tydeman Reef is the closest feature that has a name. So we're calling it Tydeman Knoll. We came up with that as we were working on the multi-beam map data. And there's a suggestion that we should name it after Rob. Well, there was a suggestion of a name that we could name it after Rob, but Rob's far too modest to have a mountain named after him. So for now, it's Tydeman Knoll.
I'm not modest. You can name it after me if you like.
Yeah. When you're in the ocean mapping game, you find all sorts of things. We've discovered lots of seamounts over the years and things like that. We can informally call them what we like. Of course, the formal naming procedure is quite a lot more involved, but this is a beautiful sea spider. Classic pycnogonid. Decent size too, good 10 centimeters across. You can see the lasers at the bottom. I think we saw one on our last dive there, Marty. Got a glimpse of
Yeah,
one.
we did. Not as big as this though. It was quite small.
Yeah, beautiful. See the sort of four prominent, you know, or 8 prominent arms, legs rather. Sea spider. If you're arachnophobe, that'd give you nightmares, wouldn't it?
It would. A sea monster arachnid. We're just positioning. We're just going to go in and take a close little look at this cup coral that's on the side of this little edge here.
Yes, we're talking about naming things. Apart from the, you know, the deep sea features, geological features we find fairly regularly, you might be surprised that a lot of the coral reefs on the Great Barrier Reef are not named. Right near us, because it's quite a remote part of the Great Barrier Reef, most of the reefs here are just unnamed. So if you look at a map, it just says UN reef, unnamed reef. Fortunately, the one closest to us here is Tydeman Reef, and hence it was quite obvious we just name it after the closest named reef, so Tydeman Knoll. And I looked up the IHO nomenclature for naming features, and a knoll is a prominent seamount feature, or a feature that's less than 1,000 meters in height.
Jeremy,
Do we collect this cup coral?
I was just about to ask you if this cup coral is something that you want to collect for your project. If not, I'll collect it for my project. But you get first dibs on this one.
No, go for it. As long as it's collected. Yeah, go for it. It's all yours.
Great, thank you.
It is a pretty one.
Jeremy, I'm just handing over to Marty. I'm going to grab some lunch.
Enjoy.
There's a question on the
Yeah, collected a whole bunch
YouTube.
of,
Sorry. No, you go ahead, Jeremy.
no, go ahead.
I was just going to say there's a question on the YouTube chat about are the red patches crustose coralline algae. So no, we're still too deep for coralline red algae, although that usually is the first colourful thing that comes into view as we come into the mesophotic zone. But the crustose coralline red algae does still photosynthesize. So we usually start seeing that coming in around 190, 180 metres. So still too deep for that at the moment. So the orangey red colouring that we're seeing here, my guess is that that's basically oxidisation at some point. So oxygen in the water is reacting with any sort of ions that are in the sediments and in the rock, and there'll certainly be iron in that ferromanganese crust, and when it mixes with oxygen it literally rusts, oxidizes. So that's the orangey red colouring that we're seeing there.
Yeah, it's funny when we collect these and then you know, preserve them, put them in a jar. The tissue, as you could see, it just retracted to inside the colony. But it certainly has the tissue, although the base of the coral is probably an important morphological feature to include in identifying the species, but certainly have images and tissue for DNA. Yeah, these guys are really hard to collect. Using the claw is challenging because they're so fragile. And sucking them up is also challenging. it can also damage the coral, and sometimes the corals get lost when they're sucked up with the vacuum. So
yeah, we're going to go back and just grab that little bit of stem as well and keep that. That'll be useful for doing isotope and trace element work on the skeleton, so we'll grab that as well.
Yep. It's a cute little sea star.
So it's just a question about water temperature. So the temperature at our water depth at the moment, we're at 417 metres depth. The water temperature is 12 degrees C, where we are right now, and on the surface it's about 26.5 degrees, maybe a little more as we continue further north. It's obviously getting warmer.
That's a cool view to have the manipulator arm angle. Looks like you've been collecting a good amount of rocks on this dive.
Yeah, just two, although they're broken, so it looks like more than two.
We need some colors to brighten up those bio boxes.
It's going to be interesting to see what communities are at the top of the summit of this knoll. Looking forward to making our way up there today.
Absolutely. I mean, the top of this feature, it could act as kind of a stepping stone between other reefs at similar depths that allow with the ranges of these shallow water species to extend across deeper habitats. You know, there are some species that there are some species that we think are cosmopolitan, which means that they're found in every ocean. But when they're not extremely deep species, like, you know, at 200 meters, how can a coral that occurs at 200 meters have connectivity between, you know, across the Atlantic and the Pacific Ocean? And the answer could be that they have these stepping stones, these seamounts, these features that come from the deep up to the shallow, and allow for corals to you know, settle and grow and reproduce. So it'll be really interesting if we can see what's at the top of this and then relate these species to their known ranges. It might help explain where we see them, where we do.
Go back and get that little piece that we left behind. It's all very valuable.
And so while we're kind of sitting down here, before we get off the sea floor, maybe it's worth moving the camera up and looking out across the, the floor above us. And that's
so you
usually
can
a good
sort
way
of
to see
see anything that's in silhouette? See, is that what you mean?
Yeah, like
look up and out.
Yeah, kind of see out across the field, for little things popping up that even when you're above it looking down, you might not be able to see it, as well as looking out across the platform. There's questions about SP for species names. And so if you're not certain about the species name, but you know what genus it belongs to, in this case, yeah, you could just say that it's whatever the genus name is, then SP. And if you have some sort of an idea of what species it might be, but you're not 100% confident, you could use CF, which would mean, compare it to the type specimen from which that species was described. So if you don't know what species it is, you'd use SP. And if you have a pretty good idea, you could use CF. And later on, you would compare it to the type material to determine whether it's the same species or if it's just closely related. Yeah, with most of the specimens that we collect, I mean, it's really unlikely that we ever really know the species of any of these corals that we're collecting. So generally everything is just SP. We might have some idea allowing us to say CF, but really it's very, very hard to confidently say what species these are, just because so little is known in this part of the world and in this depth.
Looks like we've got lots of polychaetes poking up out of the sediment here. No corals or sponges by the look of it, but we need to check.
Okay. No, it's a really good view. This is the kind of moment where you'd find one of those really little odd corals that so few in the history of deep sea exploration have been found. On the last trip, often when we're settled down for another reason is when we actually saw these interesting corals. So it is worth it to once in a while just plop down and look around, look out. Right, but these look like little, no corals of interest. What's that?
I'm just going to see whether we can get any closer to that. We're all sitting here very intently with our eyes peeled.
Actually, I did just see a view of the control room, and it's like looking down at your phone. So,
I was commenting to someone on shore about the rocks.
yeah. Hey, Marty. Okay, I can see you're looking. Remember, I always have an eye.
It's all about the eyes.
Right. Thanks to John. He always knows when to, change the view to either me or the control room. He's very good at that.
There's like some sponges in there. Perhaps. All right. Shall we carry on, Jeremy? I think we've had a good look around here.
Yeah, sure.
Yep.
Yeah,
Head.
that was good though.
Yeah, we'll keep going.
Let's do it.
We've all been just doing our lunch break swap arounds, so a few swaps and changes going on in the control room at the moment. We've just had a changeover with the pilots. We've got J-Rod piloting and Cody on co-pilot. And
Very
we've
nice.
just enjoyed a very nice Indian lunch.
Very nice.
Chicken coconut soup. Very nice. Butter chicken.
Lentil curry
Lentil
and
curry,
rice.
yeah.
Another cup coral there to the left. That white.
I think the microwave beeping was coming from Jeremy's lab. Did you have something beeping in the background there before, Jeremy?
I did not. I'm alone in the office now, so it's pretty quiet.
There's the inset showing the area we've been mapping over the last few days. It's this grey-blue area that Falkor has been working in. This part of the Cape York is just offshore of Princess Charlotte Bay, which is a very, very prominent wide bay on Cape York. And so these red areas, these shallow coral reefs, and we've been mapping right up to the very edge. Actually, if you look in the background, you'll see one of these canyons actually joins to an old river channel. Would have been a river channel when sea levels were about 100 metres lower, say at the last ice age, about 20,000 years ago. Snaked its way across what would have been the exposed Great Barrier Reef shelf to end at the head of this particular canyon. So we dived on this canyon a few days ago. We call it the Rodder Canyon. It's near Rodder Reef. See the how the old river channels that cut into the sea floor. It's preserved there now, around about 50-60 meters deep at that point. Then it drops, the offshore area drops down to over 2,000 meters deep. So it's nearly 2 kilometers in vertical height.
Some sort of little fish underneath that ledge there on the top left.
This is an Echiuran worm. So the proboscis is what's stretched out in front of us. If we were to touch that, then so we're able to just touch that briefly, you'll see it disappear back into the hole. So they typically have this sort of Y or T-shaped proboscis draped on the seafloor, capturing any organic particles that drift down and stick to the proboscis. It's going to give it a boop, and we'll see it withdraw into the hole. There we go. And we've seen some very long ones of these, meters long, draped over the seafloor. Have found them in all kinds of environments, typically muddy environments, but also in canyons, right, where it's quite disturbed. They seem to be able to exist, pull themselves in a hole if any rock material falls from above, and then they lay their proboscis out on the seafloor to capture food again.
Another sea urchin.
Yeah, we've seen quite a few. Here's another one of these Echiuran worms. You can see the proboscis just sort of hanging there. The giveaway is, if you look at the end, they split into like a Y or T shape. And, you know, we have seen them, I don't know, at least 5 meters long, probably, the longest. But these are actually quite small. So the main body of the worm is up in that hole. I've never actually seen it exposed ever. You just disappear in a hole, and I'm assuming it's kind of, you know, sausage shaped.
Oh, is Bivalve saying hello to us? We're joined in the chair here by Vicky Lowe from the University of Queensland. If you'd like to introduce yourself and say hello, Vicky.
Yeah, sure. You guys can hear me, I'm assuming? Yeah. My name's Vicky. I'm from the University of Queensland. It's far cry from my research, but certainly interesting to me, that's for sure. I'm a Southern Ocean person, so the reef is not really my specialty. Although I have to say I have done some research in the Gulf of Carpentaria, just on the western side of this point of Queensland, directly west of here actually, over at Weipa. I did some estuarine studies looking at all the macrobenthic communities over there in the estuaries, looking at them as food source for the shorebirds that come down to the northern Queensland tip, the Cape, into Cape York. It certainly didn't look like this in the estuaries, that's for sure.
Wow, that's a spectacular view. Any comments on the fish there?
What's the top of what's the depth at the top of this knoll again?
The depth at the top is 340 metres. At the moment we're at 410. So we haven't got a huge vertical distance to cover, but we're going to cover quite a long lateral distance, and then come up towards the ridge line on the edge, and then spend a little bit of time on the summit.
Right. Awesome. And hi, Vicky. It's nice to virtually meet you.
Yes, nice to meet you too.
Thanks for helping.
No worries.
Thanks for helping us out first time.
And what's underneath that ledge?
No, it's definitely good to see some things that I wouldn't normally get to see.
Oh yeah, well, me too. There's a lot. There's a lot of weird stuff down here, and it's hard to be around all of it. So it's a nice learning experience for, I think, everyone involved. Even Rob, who's been on a million of these, well, not with ROVs, but deep sea related work, and I think he still learns more and more each dive.
You really get a sense of just how steep this slope is here that we're climbing up when you get that sort of wider perspective. We're hoping that it wouldn't be completely covered in sediment, but I guess can't have it all.
Yeah. Well, it's interesting. I mean, yeah, we'll see if it does stop as we get a little bit higher. Let's see. We're getting there.
Oh yeah, there's ROV SuBastian on the inset picture there, on the side of this what we're calling Tydeman Knoll. Oh, there we go. Is that a cod? Yeah. A little bit of fish action. The white line there is the trail, the ROV SuBastian's track that we've come up the side of that slope.
There's a question about measuring the temperature. So I think the SuBastian is constantly measuring the temperature. And I assume they would pass through a thermocline. I guess Jared would probably be able to answer
Goliath
that.
grouper. Yeah, so of course we're logging the temperature all the way down. So we would have passed through that surface mixed layer and then into the water mass that we're at now. We're not sitting here watching, tracking that temperature profile all the time, although we could if we chose to, but all that data is logged and saved, and we can go back and look at it later. And certainly with respect to all of the sample collections that we make, we can then look at the data and see what depth we were at, what was the temperature, what was the oxygen, what was the salinity. So all those parameters are being recorded and saved, and we can go back and look at all that data later. Oh, look at this.
Oh, wow.
A nice little cave in there.
Yeah.
With a fish in there. Wow, look at this.
Ooh. This might be a good place to find some branching corals that probably can't occur elsewhere outside on this knoll. Let's see. Yeah, another interesting thing about having the temperature data is to try to determine what drives biodiversity as a general rule. Is it, is temperature driving biodiversity, either high or low biodiversity? Or there's a coral there. Maybe zoom in on that yellow branching. Maybe it's a stylasterid. So if temperature is driving biodiversity, or if it's depth or salinity, so having this data and also knowing exactly where the corals are living can help tease apart the multiple drivers of biodiversity to understand, what allows a place to have biodiversity that's high, and then you can therefore predict what corals live in which habitats with this data, which is something that a lot of people are trying to do now, you know, predict the extensions of different ranges of species and things like that.
Just coming in on that
so this
question
coral,
stylastrid there, if that's what it is.
Yeah, let's see. Just a quick zoom in is all we need. I mean, it's just interesting because it is in this little cave inlet, so you probably wouldn't find it outside, not with this sediment, this thin layer of organic material.
It has associates on it, Jeremy. It looks like it's got some little brittle stars entwined around on that. Is that anything that's of interest to you?
What do we think is on the top right of that coral from our perspective?
On the top right, but still tucked in under the ledge. Is that what you mean?
Yeah. It's interesting. How big is this coral that we're looking at here?
Oh, there we go. What's that? Maybe 15 centimetres, 20 centimetres? 15? Can you see the lasers there, Jeremy? They're just on there now. It's a little wider than, yeah, I reckon about 15 to 20 centimetres across.
Is it challenging to collect? I know it's kind of tucked into a cave here.
Cody can get it. Do you want the whole colony if we can get that?
Yeah, I'm curious what's attached to it. Obviously the brittle star, but also whatever is on the top right, probably attached to the base of the colony, if it's a sponge or something else. So yeah, give it a go.
And is this a regular biobox collection?
Yeah. Only because I don't think it would even fit in the quiver. Oh, yeah,
what if we took a fragment for the quiver and then the rest?
I think it's okay to just put the whole thing in the bio box in this instance, because it would be challenging for you to pick and choose specific parts to put in the quiver without just breaking the whole thing.
Yep, sure.
So I think in this case, yeah, there's a lot of stuff living on this coral. It's really interesting. We can try to, assuming there's not too much to collect. If there is, then we could then disregard what I'm about to say. But if there isn't much to collect, we can try to put this in its own bio box. So at least we know the associates in this coral associate with this coral, if you know what I mean.
Yeah, got ya.
Can you ask J-Rod what the diameter of the quiver is?
A 100 mil, 10 centimeters.
Okay.
Jeremy, if it's all right with you, can we put this into one of the small, like A or B that just has a rock in it? And then we won't put any other biota in the same compartment.
I will allow that. Nice. That's a good grab. Oh, yeah. Yeah, so we'll see if what's attached to this coral, if it is a sponge, it might be a really interesting sponge. So obviously the people on board will be able to look at it closely and separate it if it's two different types of corals or if it's a coral and a sponge.
Great.
And
okay.
Take samples of each. Awesome. Yeah, so I think one shrimp in the coral, and then of course the brittle stars. So probably a few other things we didn't see that are hiding in there. Awesome. Anything else in this little cave inlet?
Let's have another quick little look around before we move on.
Yeah, a quick one. Yeah, okay. What should do?
All right, we'll lift off and continue on our way.
So we had a good question earlier about was there any evidence of early hominids, early Aboriginal Indigenous people on Australia near here? It's a really good question because it's just the sort of thing some colleagues of mine are working on. Professor Sean Olm from the Australian Biodiversity Heritage Group at James Cook University leads a big team of people looking into just this question around, I guess, the earliest hominids, earliest Aboriginal people into Australia. And in North Queensland, where we are now, there's a well-examined caves. I think they're getting ages around 45,000 years. But the important thing is this Cape York region has been in the long past a thoroughfare, a migration path for the very earliest hominids coming through Asia, through Papua New Guinea into Australia. When sea levels were much lower, that whole landmass, Australia and New Guinea, were joined together, called Sahul, S-A-H-U-L. That was the landmass that was around years ago. So those early hominids came down through Papua New Guinea, across what is now Torres Strait. Of course, back then it was like a land bridge into Australia, and very quickly spread around Australia. So there's some work happening at the moment. Sean Olm and his team to try and understand how those migration paths would have linked together with the landscape. So we actually haven't found any underwater caves or skeletons or anything quite obvious as that in the underwater region. But if we go back to the last ice age 20,000 years ago, the continental shelf, which is only about 6 kilometres away from where we are now, high and dry. Even we've mapped evidence of old river channels now preserved on the seafloor, and Aboriginals would have used those channels for food or and forwards across the landscape. Coral reefs as they exist now would have just been flat top limestone hills with kangaroos jumping around them and growing on the sides. But now, of course, they're the sea level's higher and they're covered in organic water with thriving coral reefs on them. And just want to shout out to Tiffany C, who's watching. Great to have you aboard, Tiffany. Tiffany's a long-time fish expert, especially in the Great Barrier Reef region, although I think you're down in Victoria at the moment there, Tiffany. But of course, lots of people watching virtually from all around the world. Tiffany did her PhD studying mesophotic fish here on the edge of the Great Barrier Reef, so I'm sure lots of these fish here are quite familiar to you there, Tiffany.
Nice big urchin.
Not seeing a lot of black corals here, Jeremy.
No, I'm not seeing a lot of, most of the branching corals that we're normally seeing. I think it's because of this bit of organic material that's floating down. It seems to really be limiting what can settle and survive. But having said that, where there are little inlets, we just collected that stylastrid with associates and maybe a sponge attached to it. So where these things can grow, they will grow. So looking for those underhangs and little inlets, we'll probably find some things. There's a crustacean in front of us. And we're seeing sponges, little sponges. Not the targets that we're looking for, bryozoans.
These little delicate hydrocorals, stylastrids. Very pretty, often found in these depths. We've seen them out in the Coral Sea on those atolls out there, around about this depth, 400, 300 metres, and tucked away in caves. They seem to like to be in the dark.
I think we just passed a branching coral. Do you think that's a stylastrid we just passed?
Yep, that's right.
Okay.
So I've got a question to to John and Will down in CSIRO in Hobart. These sort of net-like bags that we're seeing attached to the walls. Now I've seen cutthroat eels sitting in amongst them. Is it possible that they're creating these bags for sleeping, or is it created by some other animal? I'm just thinking about how parrotfish in shallow waters, they can generate this mucus bag that they sleep in at night, and then they wriggle out of it in the morning. Daytime feeders. So is it possible these eels are creating these bags?
Everyone's deferring the question.
I mean, we do see these mucus bags really frequently, but these depths and even deeper. But I recall a previous dive we actually found a basket work or cutthroat eel sitting inside. It looked like it was sleeping. Okay, sounds like we're not really sure, but anyway, very curious what could be making them, because we just see them everywhere attached to vertical walls.
Will wants easier questions to answer.
Looks like an egg.
This looks like an egg casing from a gastropod snail.
Oh, okay.
Right, they often leave these sort of spiral-shaped, like little egg casings attached to a rock. Be a bit difficult to tell, but the only way to really tell is actually take a piece and look at it under a microscope. You can just make out the kind of little hole, little dots. Yeah, I'm leaning towards the egg casings from a gastropod snail of some kind.
Yeah, I think so.
J-Rod's going for sponge. It is amazing, this camera, that you can be so far out and then zoom in like pinhead sized detail. We've got nudibranch eggs, okay, so not quite hard shell gastropod, but nudibranch, yep, that sounds quite possible too. Tiffany's saying nudibranch rosettes. I like that.
That's what I thought it was too. Nudibranch egg spiral.
Yeah, me too. I knew it was nudibranch eggs as well.
Just looking at the rock face, Rob, you can see how, I mean, there's the dark ferromanganese crust on top, and it looks like these are all just cutaways where chunks have come off, and you can see that it's a fresher face with the crust overhanging, so it looks like there's big chunks missing there.
Yeah, so if it's stratified but layered down, so we're not talking horizontal layers, you know, like the layers of like a wedding cake. Imagine it curved around the edges, and looks like the pieces of that shear off. So we are looking at layers of strata. It's the real key is to get some of that rock sample. Hopefully we've got enough that we can look at it hand lens and look at its composition. We're definitely leaning towards this being an ancient drowned reef preserved here on the seafloor. Just quite strange that it's kind of sticking out from the edge of the Great Barrier Reef. You know, in the long distant geological past, this would have been shallower, closer to the sea surface than, for whatever reason, maybe subsidence. Its seafloor has dropped, and whatever coral growth was at its surface has not been able to keep pace with any relative change in sea level, and then it becomes drowned. And so it's just sitting here as this preserved hard edifice on the seafloor, now being draped with this soft sediment and the hard rock that these deep water animals can attach to.
There must be a huge amount of material that has eroded away that is now not there. We're not seeing it. All we're seeing is the remnant, sort of the most resistant, I guess, cemented, calcified remnant reef rock that's left. This must have been quite a much larger feature, and most of it now is actually eroded away.
So there's the SitCam inset view. This is the widescreen view. The ROV's got about 10 cameras all around it, but the main ones we use are the science camera, which is the one that is mainly streamed on live. The situation cam, which is a lot broader, a lot wider perspective, and that gives us a larger view of what we've got around us. Jeremy, I understand you're probably going to log off fairly shortly. Is that the case?
Yeah, but I'll continue watching. I think as we get to the top of this, we might expect more to look at and potentially sample. So I'll be chatting over Slack.
Okay, no worries. All right, Jeremy, thank you very much for joining. So Jeremy's going to sign off from Townsville in Northern Australia, and will bring us back to the control room here on the Falkor with just myself, Robin Beaman, and Marty McNeil next to me. We'll take you through the afternoon on this dive on Tydeman Knoll. Okay, thanks very much, Jeremy. Catch you later.
Thanks, Rob. All right, talk to you guys.
That is steep. It's a lovely perspective to zoom out and get a broader view. Thank you, J-Rod.
Isn't it
It's
steep?
amazing. It's very steep. But yeah, surprising that there's not more attached to it though.
Yeah, I guess we were hoping for a lot more of these more dramatic, large colonies. There's certainly a lot of sediment draping over the top of it. It really needs a steep vertical wall to get any kind of attachment. But most of what we've been seeing is sponges, solitary cup corals.
Polychaetes.
Yeah, polychaetes. No shortage of echinoderms. Lots of sea stars, fairly large urchins, but no real big, no big scleractinian coral colonies or black coral colonies. We're still working our way across the eastern face of this big knoll. Lots of little white cup corals poking out.
Looks like a nice stylosterid sticking out on that
Yeah.
folder there.
It's quite a strange shape, isn't it? It's just sort of sitting there like that.
I was imagining in however many million years ago when this was the living reef, you know, picturing some kind of what coral morphology would look like that.
You were thinking that that's like some coral colony sitting there, stuck on the side. Looks like it's a great seat. It's like
Yeah, and there's
a
someone sitting on
throne,
it.
throne fit for a
For a shrimp.
for a shrimp.
Oh, avalanche.
Yeah, the mud definitely suppresses what's living there, doesn't it? Like you're only getting most of that life on those vertical surfaces. But we do see this in other parts, like at Osprey Reef where we visited, really the steep sides. There was so much calcium carbonate, limestone rock, and algae raining down from above. It was just like this constantly bulldozed slope. So there actually wasn't a lot living there on the surface, except where it was steep enough that you could, that the sediment didn't accumulate, and then you got lots of animals attached.
And there's the view looking outwards. A lot
Pretty
of
steep.
particles in the water.
Yeah, yeah, but you only need a little bit of a gradient, and it just seems to paste itself onto the surface.
What's that? Is it sponge?
Yeah, it looks sponge-like.
Interesting morphology too, isn't it?
Yeah, when you ducked out for lunch, there was this sort of split in the rock, and it had all this dark ferromanganese oxide sticking up through it, like a
Vertically.
yeah, vertically. Trying to think how would that form.
Yeah, I saw that when I was sitting down in the crew mess having lunch. It looked like a joint was secondary, and then that vertical infill was a secondary fish
What do we got here?
mineralisation that's precipitated and filled in the joint.
Is this one of these slime heads? Looks like it's had someone's had a bite at it.
It does.
On the side, on the left-hand side. Roughy.
Roughy.
Yes.
Ah,
Yeah. Not the prettiest looking fish. If it turns around for us, you can see something's had a go at it on the side. It's a good look at it. Whoa,
there you
there you go.
yeah, it looks
Is
recent
it?
too.
Yeah.
Interesting.
Yeah, we have seen roughies out in the Coral Sea. Oh, nice cave. We want a few more of those caves. Yeah, John's saying Gephroferix. Gephroberix, if I got that right. It's a little cardinal fish in front of it.
Yeah.
Yeah, okay, so we need to look for these caves. They're clearly great places to find fish. Epigonids. We're going to duck down and have a closer look in the cave. great spot. Great spot to hide.
That's where I'd be hiding if someone
Yeah.
was trying to take a chunk out of my side.
Yeah, or some giant alien-like creature came out with lights and big waving arms. That's where I'd hide.
Yeah.
Yeah, thanks, Will. Jeffra Barracks. Getting a bit tongue-tied over my Latin. Ah, more caves. This is good. You notice these, they look like bivalve parts on the rocks. There's not, I don't see too many live ones, but the one half of the shell's still cemented on the rock. So they must have been quite numerous in the past.
Look like oysters almost.
Yeah,
Don't they?
just the remains of oysters.
Another nice little overhang.
Yep, fishing them. Oh, look at this. It's a rock.
They drop off.
Yeah. How sick is that? So it looks like we've got a feather star coming out on the side. One curled up. A few of them curled up.
Yep, there's a few more coming in on the right there.
30?
This looks very karst-like to me.
It does,
You know, that
doesn't it?
eroded limestone karst surface.
Yeah. Of course, the more nooks and crannies there are, the more surface area there is for other things to colonise. But yeah, not seeing as much as I thought we would on these surfaces.
Yeah, but you know, they become little micro-habitats. And we've seen this out in the Coral Sea on those Queensland Plateau atolls. We know they are ancient atolls themselves, and so where you have these exposed faces, you get the layers, the different strata layers, creating these nice little individual microhabitats. And you know, that's clearly what's happening here too.
Nice urchin. Yeah, this limestone is very clearly very old, very eroded, weathered.
Yeah, in that case, it probably has more in common with those offshore Queensland Plateau atolls, which are millions of years old. You know, formed during the Miocene originally, and that's possibly the case here too. So north of us we have these strange detached reefs. Rain Island is one of those reefs. They're much older than the Great Barrier Reef itself. As far as we know, Great Barrier Reef formed on the continental shelf. We've got kind of origin ages back to around about 500,000–600,000 years ago, which isn't very old in geological time. But then we have these Queensland Plateau atolls and reefs which formed in the Miocene, much older. And my guess is we're looking at something here that's more similar to these offshore Queensland Plateau atolls, probably with an origin in the Miocene, so millions of years old, as opposed to just hundreds of thousands of years old.
That's right. And I think one of the things we don't know, or I don't know, you might have more of an idea, Rob, is the timing of the emplacement of that ferromanganese crust. How long does it take? How quickly does it form? Those sorts of things. I have no idea.
Yeah, you need surfaces to be exposed to seawater for a really long time. You know, in millions of years, and as long as it's a stable environment, and that rock is exposed to the seawater, that ferromanganese crust can build up. So we did an expedition last year further out in the Coral Sea, and we were picking up pieces of rock from the seafloor. It turned out it was originally volcanic rock. Oh, there's another one, Echia and worms, big long proboscis. You see how it splits in this sort of classic Y shape. And when we picked up the rocks, they had, in cases, up to 10 centimeters thick of this ferromanganese oxide coating around the nodules. I mean, people know them as deep sea nodules, and they mine them because the ferromanganese oxide concentrates rare earth metals like cobalt, even gold, within the oxide crusts, and these nodules can coat the deep sea floor. And so today commercial companies are actually mining the deep ocean floor because of these nodules are concentrating precious metals. But here we actually are seeing the crust, that black crust. And it would have been millions of years. Oh, we've got fish in the background. Let's have a closer look at this. Yeah, I'm seeing at least three.
Yeah.
3 fish. No, I'll definitely defer to our fish experts. I was going to say trevally, but they're not. No, I don't think so. Beautiful, though. They are really pretty. Beautiful gold shine on them. Got jackfish, jobfish.
Here we go. All the suggestions are coming through. What's Will saying?
What's the common name for us? For us on board there, Will? Beautiful. Look at that.
Does look like a snapper.
Barramundi.
Ruby snapper.
Ruby snapper. There we go. Thank you. Yeah, we saw at least 3 of them. We're at 430 meters. It's pretty well deep, but maybe not deep for these fish. Yeah, Daniela, ruby snapper, beauty. Good to have Daniela joining us. Danny is one of our principal investigators. I think are you in Townsville, Danny? Wow, very curious. Look at the teeth on it. Yeah.
Beautiful.
Yeah. Danny's at Magnetic Island. Yeah, I took my family there for a holiday just a couple of weeks ago. So Danielle is our resident reef ecologist. possibly a bit deep for where Danny's expertise is, but she's joined us on previous audio. For information, we will be doing shallower dives on the edge of the reef as we get further north. So we'll definitely be in mesophotic zone country, and hopefully right up into the shallower parts of the upper slope of the Great Barrier Reef.
It's like this urchin is just hanging tenuously onto the side of the face there.
Yeah, big pencil urchin.
Is it? Cliffhanger urchin.
All right, now I think these are worm snails. Yeah, so not some. It's probably a mix. There's some some polychaete tubes, but I think the larger ones are worm snails, and that's from a German expedition that came out here in 2009, further offshore at the Osprey Reef and the Queensland Plateau, and I was misidentifying these kind of large white tubes as polychaete tubes, and Gert Vorheid was the chief scientist, and he's going, no, they're worm snails. So they're a gastropod snail that create these, that cement these fairly large tubes against the vertical rock faces.
Nice bivalves in there. When we're looking at reef rocks and we make thin sections out of them, you can tell the difference between the worm tubes and the gastropod tubes under the microscope when in thin section, because the structure of their walls is different.
Oh yeah.
So
Making that very thick crust. I mean, it's quite obvious, isn't it? But it seems to be harder than what's underneath, and underneath's eroding away, but it leaves this hard crust.
Yeah, the
on
crust
top
is more resistant.
So we're doing a long sort of almost horizontal transect across the face of this Tydeman Knoll with the aim to get to the crest, which is still about 100 metres or so away from where we are now, and then we'll be on the top of it, the sharper point of the crest, and then we'll work our way up to the summit. And yes, the inset is showing us how we're traversing along the face. So imagine that's like a small hill. It's about 1.4 kilometres along the base and about 500 metres wide. And we started at the right at the base of it on that fairly gentle muddy seafloor. We've climbed up about halfway across the face, and now we're traversing around about the same depth, with the aim to get to the crest, which is right in front of us. Some great, excellent graphics there. And then we're going to work our way up to the summit, which is around about 340 metres depth.
And who knows what we'll find there. Probably more of the same, but
could be just more mud, more sand here, Marty. But you never know unless you look.
You never know. You've got to look.
That's right. Yeah, depth of the summit's about 340 metres. The western side has, it's slightly more rounded, and it seems to have more like some gullies on the western side. But we chose this eastern side because of where the wind is. The wind is coming from the southeast, so the ship needs to point into the wind. So we're in a good spot as far as the ship goes when we deploy the ROV. So we have some real limiting factors as to where, you know, we have to choose very carefully where we put the SuBastian. And so part of the, you know, the discussion beforehand, like the day before, we all get together as a team. We talk about what we'd like to do, and then we bring in all the environmental factors to say whether it's doable. And we always have a plan B and a plan C if when we get to the site we find that the weather's not what we need.
Yeah, if we were on seamounts in the open ocean, it wouldn't be so many factors to consider, but we're so close to the reef here that it's a lot of factors that we need to take into account. As you say, Rob, with the positioning of the ship and the wind, the waves, the currents, and our exit strategy. If we need to make a quick runaway, we can't be facing towards the reef.
Yeah, that's right. We need plenty of sea room to do this kind of work. You imagine, you know, the ROVs in the water. We've got the ship tethered to it through a big cable. You need a lot of room to manoeuvre.
An anemone, a nice anemone.
Yeah, and what about in the background here, Marty? What are these? Are these stylastrates or?
Stylastrates. Yeah, let's go there and then there. That's a nice anemone.
Big anemone. Haven't seen any with clownfish in them yet.
No, definitely not.
That's a classic anemone. What a beauty.
Beautiful.
You know, they'd be good, what, 20 centimetres high?
10 centimetres across at the base.
Yeah. I wouldn't like to touch it. I'd probably give you a nasty
Nice
zap.
sting.
Yeah. Yeah, I've been hit by anemones in the past. They can leave a burn on your skin. Yeah. Something living on top.
Beautiful colours, aren't they? Oh yeah.
It's like your classic textbook Cnidarian, right?
Yeah.
Whenever you see the textbook example of what a Cnidarian looks like, it's usually like a split in half one of these, with the mouth, you know, a stomach, the side walls, tentacles. Oh,
Here
shrimp!
we go. There's a little shrimp.
Little commensal shrimp in there.
The shrimp has eggs. The shrimp has eggs.
Oh yeah, under its bottom, its legs underneath.
Wow.
Same colour as the anemone.
Yeah.
Yeah. Perfect spot.
Beautiful.
So we're joking about clownfish or anemone fish. You know, here we have an anemone shrimp, just making its home on top of the anemone, and obviously immune to any of the nematocyst stinging cells on the tentacles.
Beautiful.
Yeah, so Nini's asking, do you think it's evolved? And I think yes.
You'd have to think
it's got
so,
the same
wouldn't you?
colour.
Yeah.
You know, it's obviously using it as a home, as a habitat.
Yeah. Otherwise, the mouth on that anemone could just swallow that thing whole.
So it's curious, does the anemone get any benefit by having the shrimp there? But you know, this is sort of an open question with like anemone fish, for instance. You know, anemone fish can push away or attack predators that might, so that might be an advantage that the anemone have. Or, you know, could they attract food, you know, like, hey, I'm a shrimp, come and eat me, and it brings food in towards the anemone. So it's sort of interesting, these ecological relationships, but it's not always clear, you know, if it's an exact two-way relationship.
Yes, yeah. Cleaning could be another thing. The shrimp could be cleaning.
Yeah.
On the outside. All right.
It's a beauty.
That's good to go. Go and check out those, whatever they are, to the left. Yeah, there we go. Those guys. Look at these now. What are these?
Just going to have a closer look at this fringe of coral, get a sense of what we're looking at here.
This does look interesting.
All right, any guesses?
1, 2, 3, 4, 5, 6, 8.
8.
Tentacles.
So it's soft coral.
Octocoral.
Yeah, octocoral. We've got a few gastropods in amongst it. Hermit crab, Stylastera down here.
Yeah.
But what kind of octocoral? Anything from Jeremy? Nothing?
Okay.
So we're just reaching out to some of our other principal investigators ashore. We're in direct communication with them. They're watching just so that we know whether to sample them, and if to sample them to go into the bio box with which would be preserved in ethanol, or to preserve them using liquid nitrogen, and that's to get their full DNA, full genome structure.
Quite a colony, isn't it?
Yeah. Yeah, I don't recognise this.
No.
I can't say I've seen these before. All right.
All right, well, we might carry on.
We'll carry on then.
Yeah.
Yep. Haven't heard from our investigators ashore, so we'll continue on. Looks like the summit to me there, Marty. Must be getting close. Yeah, we've cut across. So that would be the summit up ahead.
No, it's fine. Yeah, so it looks like we're on that ridge, top ridge.
Some more anemones, perhaps.
Yeah.
So think of this drowned reef like a big, low hill, big, low rounded hill. And we're right on the crest of it, so we're going to walk our way up the crest, see what we can find, up to around about 340 metres depth.
These anemones look like boab trees or something, just sticking out like that, don't they?
Yeah, there are more fish. Some of those ruby snappers in the background. Pretty shy. So we're at 386 metres. The summit's around 340, so it's not too far vertically above us now. But we're just going to walk our way along the top, just see what we can find.
Updated waypoint bridge. Update, thanks. Thank you, Eric. Thank you. Good, awesome. Your hair looks nice. Your hair looks nice today.
No, thank you, Cody. Thank you.
You didn't say you have nice hair. Do you have nice hair, though?
You have nice hair.
Yes.
Got our Texan crew here. Very comfortable in the ROV seat. Cody and J-Rod.
I liked your haircut at the end of the last trip.
I know, me too, but
You didn't want to keep it? Extend it?
it was like floating right here.
Oh no.
It's not growing like her. I'm losing my hair. At 28, so throw again or something. Don't have to have.
Oh, I've seen these before. They're a shell, and they attach.
Gastropods on it?
They attach to the side. Yeah.
Oh,
Let's have a closer look at this.
that looks like a gastropod.
It's really odd. Yeah. Yeah.
Shells on shells.
Shells on shells. And it's even got,
got a cup
little
coral
cup
on
coral
it.
on it.
Wow.
Carrier shell. That's it.
Carrier shell.
Yes.
Xenophora. Oh,
Yeah. Very weird.
it is weird.
And it's cemented gastropods into part of it.
Yeah, okay. I see it. Don't see it.
Yeah. Very weird.
Yeah, we're good.
A little bit. Looks more like a mushroom. Like Mario mushroom.
If you see a nice crusty overhang where you think you can break off another rock from the top, that would be awesome.
Okay, there's going to be something up in the bow.
Yeah, still pretty muddy here, isn't it, Marty? Well, you know, the gradient's getting a bit more gentle,
Yeah, we're on the top of that ridge line now.
but we're looking for any real obvious small cliff that we can break off, so we can do age dating on it and look at its composition.
Like that.
Yep. Anything that you think we can.
See some of the features in the sonar as well.
Oh yeah, yeah. So this insert is the scanning sonar that we've got. It's looking ahead of us. So it's just swinging from side to side. You can see these kind of like little ridges. They leave these pock marks on the seafloor, and the sonar's picking them up quite well. And just a shout out to my brother, Greg Beaman, watching from Kia Ora in the Sunshine Coast in Queensland. Great to have you join us, Greg. down here, 379 metres in the far northern Great Barrier Reef. What's underneath? I wonder if you could
Try for the drill.
get a drill here.
Can get bumpy.
Okay. There's nothing in 1D, is there? Nothing in box D? D? There is or there isn't? No. Yeah. Should we try and go back in?
Looks a bit,
It's very eroded. It's so
yeah.
altered.
Just wondering if we could get the drill out and do a bit of drilling.
Do you want this?
Yeah, we want that too, Paul.
Yeah.
Where would you like that to go?
D. Please. Thank you. Could we try and drill, please?
Just back in that same spot.
We'll try and get what was underneath that.
Yeah, that if we can get some of that fresh face, that'd be really good. It'll be hard though, real hard.
Yellow.
Brittle start having a walking away in the background.
Oh yeah. He knows something's up. He's getting out of the way.
We're going to try and do some rock drilling.
Sebastian style.
Yeah. Just pull out the Swiss Army kit. There we go. So this is a little drill bit that J-Rod and the team put together. We used it quite successfully on the last expedition, didn't we?
Yep.
Yeah, we actually got a piece of the ancient continental rock which sticks out, a bit of Australia that sticks out underneath the Great Barrier Reef. Okay, not quite lined up. So what the guys are trying to do is come in, you know, right over the top of it, so that it's the handle part is lined up exactly within the length of the arm. So as it's turning in a nice neat circle and not wobbling around. That's pretty good.
You want us to go straight in right there? Or?
Straight in right there. Where?
We're trying to get what's under the crust.
Yeah. So this
whether you
face,
can go in on the side face,
possible.
or I'm not sure how your angle is going to work for you.
Polygons. You want to go to the right or to the left?
Probably to the right.
The right.
So what we're looking for is a bit of that rock on the steeper face, not necessarily the crust, which we've broken off a fair bit of crust during the transect so far. We're trying to get in. Here comes a fish for a look. Looks like a ruby snapper. There we go. And we're going to try and drill into that face that's just to the next to the brittle star.
The name of that knot, monkey fist, has been around forever. It's been around forever, right? Yeah, hundreds of years.
Yeah. So if you go into knot tying
Nothing
books,
new that we've come up with.
Yeah. If you go into knot tying books, you can find out how to tie your own monkey's fist, and they put them at the end of lines for throwing, like throw lines and things like that. But really useful here. So all of these little tools have got a monkey fist, so you can grab them easily. I'm going to see if we can kind of drill into that face.
Oh, right there, Marty?
Yep, that'd be great. Yep.
Okay, let's see what happens.
See how we go.
How hard it is. Limestone's pretty tough.
Yeah. It's calcite cement holding all of that together.
Yeah. Now it's got teeth, and it's just got to kind of grip it. Oh, looks like it's grabbed. Yeah, that's looking good.
Mineralogically, calcite's one of the softer minerals. It only has a hardness of 3 on the Mohs scale of hardness that we use for when we're identifying minerals. The scale goes from 1 to 10, and 10 is diamond. Calcite is only 3, so it's pretty soft. You can just about scratch it with your fingernail.
Yeah, but you
when
know,
it's in this
300 meters
ancient
down,
reef environment, and it's been under depth and pressure for who knows how many millions of years.
yeah. Then you're trying to drill it out. It's
very hard.
can be quite tricky.
It's very hard.
Yeah, no, we did try out in the Coral Sea, and it was just bouncing off it.
Yeah.
Let's see what we got.
Cleaned it.
Cleaned it, like ground it off.
Yeah. At least that shows us anyway what's in there.
Oh, it is actually cutting
So
around.
you can see the
Yeah.
cutting away.
Yeah, so it's biting into it.
Yeah.
You can see lots of little holes and things, so it looks eroded. It's not the best angle, of course.
Brittle sawmill.
It looks like it's well, from what we just got a glimpse of there, it looks like it's grains, clastic grains, but carbonate, rather than reef or framework. I guess it's a bit hard to tell until we see a bit more of it.
Yeah. So when you do, you know, terrestrial geology, you're always trying to snap off a fresh piece, you know, so...
And lick it.
Lick it or look at it on a microscope, but, you know, a geology pick is one of your best tools, isn't it? You just try and smashing off beyond the weathered crust.
Yeah, that's right.
So if we think of this ferromanganese oxide as the weathered crust on the outside, we really want to get to what is the original material, and hence it helps to actually drill it or break it off, because it's not easy to do when you're this far down. But you remember the actual ROV is actually neutrally buoyant, so the guys have sat it on the seafloor to give it a bit of purchase. And I got my sister Jody watching. Welcome, Jody, from Cairns. It's definitely chewing into the rock. But not a lot.
No.
If we get any big pieces breaking off, Marty, we should just grab them and
for sure.
Yeah.
Just grab it and go.
Pretty tough.
Very tough. Yeah. But that gives us a good look at it.
Yeah.
It might be
Lots of holes, lots of
good as we do.
bioerosion. It's definitely, you know, limestone, obviously,
Yeah.
but not enough to get a decent sample of, unfortunately.
Yeah.
All we're doing is just polishing the edge of it.
Which still gives us a glimpse of what it looks like underneath that crust. So.
Yeah, but you notice all the little holes?
Yeah.
I'm wondering if that is reef or framework, or is that just bioerosion?
It's hard to tell, isn't it?
Yeah. I don't think we're going to be able to do this there, Jaron. I don't think we're going to be able to get a piece. We're just polishing.
What do you think, Cody?
Try to go straight down on it.
Give it a try.
Yeah?
Break away that top layer and then just cut it.
Okay.
Yeah.
All right, we can try that.
Try.
Yeah. All right, we're going to try in from the top and see if we can kind of snap off that ledge, but it might give us a cleaner surface than what we've got previously, which is just snapping it off with the claw. No, there's no center drill. It's actually hollow on the inside. So the cutting part of it's just on the outside that you can see. All right, let's see if we can get through this stuff. Okay, looks like it's cutting into it.
Yeah, we've got chat feeds full of insects.
Try to drill a hole saw without this in the hole. See what happens to your wrist.
Lots of positive encouragement too.
Looks like we've just been polishing it. All right, we may just have to break it off. Is it possible to just?
You want to break it off now?
Yeah, it doesn't look like you're going to get much deeper than that. Yeah, it's a bit of shaving. Yeah, just how hard it is.
Yeah.
You better keep drilling.
We're just polishing it.
That is hard, isn't it?
Chris, you want to go grab it? How do you want to do this? Break it off?
So what do you want to break off? The crust stuff? Or
Yeah. Well, can you get your get the claw in and from underneath and kind of lever it? Maybe there's
the
it's
lip?
we can.
Yeah, we already got a piece of the lip. We can get
Yeah,
that.
let's grab that lip.
All right, right where it was drilling?
Anywhere along. Right? Yeah. Anywhere.
Yeah, it's proving much tougher, but we have had this experience before.
Box 2 open. Yeah.
Anywhere?
Yeah, just anywhere that's easy. Thanks, Chris. It's just too tough for the drill.
Yep. Rock wins again.
Mm-hmm. Well, it's been there for probably millions of years. It's not going to give way real easy.
It's there because it's resisted every other
Yeah.
thing that's been thrown at it.
Yeah. Cyclones and, you know, time. Huge amount of geological time. Many sea levels rising and falling, but still here.
Wow, what it gave, it gave.
Very tough. Okay, well, we can make use of this.
Grab that.
Put it in D was the
Where is it?
put it in box D.
Yeah, grab that.
Okay. Who is the person that makes a drill hand on a ship? Well, it was a combination of great ideas from a few people. On the last
tungsten.
expedition, we were talking about how we were going to try and get these rock samples. I think we were having a barbecue out on the outdoor lounge, and I said, "Oh, do you have any kind of rock drill? Is it possible to get a rock drill? Does Sebastian have a rock drill?" And of course, J Rod replied, "Oh yeah, we can do that."
Yeah,
so
yeah, you've got a big workshop on board.
between J Rod and the engineers, they made it for us.
But we will be able to make use of these pieces, even though it's not as fresh as we'd like it to be. We can see the composition of it even under a hand lens. You can see what they're made of, so will still be useful. Nah, I think we're good.
What's that?
Did you see that break off?
Yeah, it broke off
Okay.
with the piece that
Okay.
when Chris broke it off, that extra bit came with it. So it definitely came with it.
Lots of good advice coming from shore.
Alrighty.
Okay, let's head on up to the summit. Whoosh.
And with that, we were gone.
Well, we gave it a good shot.
Yeah.
Yeah.
Right,
The urchins?
Little
Yeah,
urchins.
quite a few of them.
Yeah.
Just okay. So we're climbing up the summit of this big hill, up to around about 340 meters. That's what we measured it with our mapping.
They're anemone.
Bridge ROV, just move yourself a little bit. Yeah, wait, thanks. Still there.
Whoops.
It's the top.
See Brisington sea stars, these pink sea stars, they're quite pretty. So these are asteroids, not brittle stars.
Right.
Some more of those soft corals that we've seen before. Remember that big fringe of white
Yeah.
white corals?
Yeah. That looks
Fish.
pretty. Is that a
Fish in the background.
Nice pink urchin in the
Yeah.
foreground.
There's another roughy.
Another
There's a few of them there.
Outcrop there.
Roughy in front of us.
Yeah. Let's pick up that rock.
Challenge accepted.
Nice. Nice little cave.
Yeah.
Oh, lots of little feather stars underneath. They're all fringe of feather stars here.
Some structure here. Can you zoom in there?
Oh, we got ourselves a shrimp party.
Shrimp party.
Yeah, I do like this. Yeah, lots of them in here. Look at them all under the rock. Heaps there.
They look like they're having a great time.
Very cute. I remember on the previous expedition we went into a vertical split in the rock out in the Queensland Plateau there, and they were all over. They just completely filled the crack, all looking out at us.
All oriented the same way.
Yeah, this beautiful sort of pink orangey colour.
The characters, aren't they? Yeah, yep. Stars and stripes.
Oh yeah.
Yeah. And there's a fish still hanging around up there. Yep. Very nice. Thank you. What's down there? What is that? What is that?
Fish.
Fish?
Yep. Having a sleep perhaps. There's more of the shrimp. There's a lot of shrimp in there actually.
Yeah.
Yeah, they're all on the side
On
of the
the
rock.
sides. Oh, they know that we're here.
Yeah.
I won't do it because there's no time in my life, even in that instance, I want to be prepared to frank up. So,
There we go.
One of those
Shells, what
collector
are they called again?
shells.
Collector shell. Who's this little guy?
Carrier shell.
Carrier
That's
shell,
right.
yeah.
Not collector shell.
What's on top of that?
Yeah. Strange.
Urchin, or is it?
Ooh, that's interesting. Looks like a banner fish of some kind.
Yeah.
Boarfish.
Antigonia.
Yeah, okay. Oh, good.
Antigonia,
deepwater
deepwater
boarfish.
boarfish.
Yeah, good. Thanks, Will.
Don't make me come back there.
Oh, let's have a closer look at this rock. There's lots of Brisington sea stars on the other side of it. And again, lots of this sort of bivalve clams, oyster shells.
There's a big collection of them here, that's for
Yeah.
sure.
Hermit crab on top. Brisington sea stars.
I'm going to Google that right now.
Look at that.
Brisington.
Brisingid sea stars.
Brisingid.
Beautiful. Oh, what have we got here? What's that? Just it's a
That's
little
that looks
slipper.
like a
It's a slipper
bug.
lobster.
Looked like a
Yeah, it's a little slipper lobster.
Brisingid sea star.
Ah, this is pretty. It's very pretty. Yeah, well, it is, like a Moreton Bay bug is a slipper lobster.
Okay.
Yeah.
Were you on board when we had Moreton Bay bugs for dinner on the
No,
previous week?
no.
They were fabulous.
Yeah, but
The description on the menu was flat-headed lobster. Flat-headed
yeah,
lobster.
right. But they're Moreton Bay bugs.
Moreton Bay bugs,
That's what we call them in Australia.
Yeah.
So you can go to a restaurant and order bugs, and if you're not familiar, you think, what?
Yeah.
Am I eating some insects?
And I haven't eaten them for years, but these were very, very sweet.
Yeah.
Really good.
Yeah.
The bugs.
Moreton Bay bugs. Yeah, one of them. There's one of those interesting shells again. Ooh, there's a big auger shell. Just left. Just pan left here. Thanks, guys. This is an auger shell. Yeah.
It's like a carrot, doesn't it?
Order A-U-G-E-R.
Yeah, very sharp.
So is it a scaphoid as well? Is it open at both ends?
It's a
Is it type of
gastropod if it's
scaphoid or
if it is
gastropod?
an auger. Yeah. Sea carrot. Yeah. Oh, I could be wrong. It could be a part of a pencil urchin spine. You get these very long spines. Hard to tell. No, I think if it was an auger, it would be more spiral. This has got stripes along the edge.
My guess is scaphoid, but we'd need to see the narrow end of it to see if it's open as well.
Yeah. We don't really know.
Yeah.
I thought it was an auger shell. I haven't seen them with this sort of vertical long stripes on the side before.
Pick it up.
Want me to pick it up?
Please.
All right.
Not to collect, but just have a look at both ends. It's open at both ends and tell us what it is.
If it's rounded at the bottom, it'll be part of a pencil urchin,
So,
Let's grab some water.
yeah. It's really hot in here. Let's check it out. So I've been saying scaphopod, which is the scientific name, but tusk shell is the common name. So we'll just have a little look at it. We'll have a look at both ends. It's open at both ends now because the tip broke off.
Sorry.
That's all right. It wasn't you. I think it might have already been broken. Yeah. So just the end of the little tip. Yeah. I'm going with tusk shell, scaphopod.
Little carrot.
A carrot, tusk shell. So, hello carrot. Is it in there? Cool. I've never seen one alive. I've only ever seen the empty shells.
You want it?
No, we don't want to keep it. But you've satisfied my curiosity. Thank you.
Here for you.
I'm glad.
Gently sit it back now. Before you get out of here.
All right, let's keep going. So we make our way just towards the very summit of this knoll that we've been climbing on today. We're at 360 metres below sea level. Oh, there we go. Is that another roughie? And the top of this feature that we're on sits at about 340 metres, so not too far to go. We had no idea what we might find up here. We could see that this feature in the mapping was very prominent and very hard, and hard surfaces tend to be where we find corals attached, Gorgonian fans, black corals. So we were hoping to see some of that, but not today. Here's a nice little cave. I suspect there might be a shrimp party going on in that cave. Mm-hmm. Yes, hard surfaces make for happy scientists. There's lots of sedimentologists that study mud, so there we go. Shrimp party. They look like they're at a rock concert or something in there.
Turn it off, turn it on. Leave it.
Rock on shrimps, yeah. Dancing on the ceiling. Fishy fish. There we go. What's that? Need to keep our eyes peeled for any pelagic fish aggregating around this prominent feature where it's about 6 k's offshore from the shallow part of the Great Barrier Reef coral reef. And this is the most prominent geological feature in the area right where we are right now. It's that little guy. Do we have a fish ID on this one?
Big eye fish.
Big eye fish? Big eye flatfish? Deep water flathead. That is deep water for a flathead. Bimbrops. Thank you, John. Thank you, Will. Carry on our way. Oh, great. Fantastic. We just got Valerie joined us in the chair here towards the end of this dive this afternoon. Valerie's one of our postgraduate master's students who's sailing with us from James Cook University in Townsville. Valerie, do you want to introduce yourself and say hello and tell us what you're studying at JCU?
Yeah, hi, I'm Marty. That was pretty much exactly what I'm doing. So I'm a master's student in my final semester right now, so I'm conducting my own research project. I'm focusing on remote sensing coral reefs, looking at hyperspectral data. Instead of really looking at imagery, I'm actually trying to find a cheaper way of doing it. So using consumer-grade drones and more affordable hyperspectral sensors, so looking at point data to try to map out coral reefs. And I'm doing so by using spectral unmixing techniques. So if you have, a point spectra, I'm actually finding ways to try and unmix the spectra to see the contribution of coral spectra in it, or algae, or sand, just to find a more affordable way for people to map out coral reefs.
When you talk about spectra, are you talking about the wavelengths?
Yes, so looking at the hyperspectral data, so at each individual wavelength you get multiple reflectance readings, so you actually get the whole spectra. And getting so much information, you can actually extract the proportion of, it's called N members, so pure N member of coral. You can actually find different averages of different species to kind of find a representative coral spectra and unmix this out of the readings that you get. So yeah, it's...
Whereabouts have you been doing your field work?
So I actually haven't been doing the field work. I'm working with Dr. Karen Joyce, where she actually collected all this drone data from Heron Island, and she's got this huge database of data that just hasn't been used. So that's why, similarly to actually Joan, she's focusing her project on sea cucumbers, and I'm looking more into actually just mapping out the reef to get a classification of different components. But unfortunately, I can't actually get to the species level of corals, so it hasn't reached that point yet.
What about morphology? Can you tell?
We have another girl. We have another girl who has been trying to do that, but so far has been unsuccessful. That's not really what my project is focusing on, though. But I know that people are trying to look at more of an object-based classification. Whereas, as I said, mine is more, I'm trying to look at a sub-pixel level classification, so trying to take this point and classifying it, but seeing how many components within this one reading you can actually get. Yeah. Yeah, it's...
Here we go.
Oh, wow.
There we go.
They remind me of coelacanths,
Yeah, they look ancient.
Yeah, but they found coelacanths in caves, in volcanic caves, and
It's like living fossils.
Wow.
Just popped up out of nowhere.
Fantastic.
It's a question about ground truthing, your sub-pixel unmixing.
Yes,
Yeah,
exactly.
convention.
It's very hard. So yeah, actually getting back to that ground truthing, I am actually in the accuracy assessment phase of my
Right.
project, and that's where it gets a little bit hard because obviously since I was not able to collect the data myself, we don't have in situ data that can overlap very accurately. So
And that's from Heron, you said?
yes,
Yeah.
from Heron Island.
I guess one advantage about having your data coming from Heron is that it's so well studied. It's been
Exactly,
There's
so-
been a research station on
yeah,
there for years. and
Exactly.
so-
There's so much data out there, and
Yeah.
it's also kind of the ideal reef to study just because it's so shallow. Usually the water is so clear, so you don't have to do much pre-processing. Wow.
A group of groupers. Or
Yeah.
gropers.
Gropers.
Meaning
Groupers.
Group of groupers.
Formerly.
Yeah.
Groper.
And they're saying bar cod.
Hardcod.
Do we know how big these are? Is it possible to get the
Just give them
lasers? It's a great shot.
Wow.
10
There you go.
centimetres.
So
That's a
the
big
distance
fish.
Wow.
between those two
Yeah.
lasers is 10 centimetres.
So,
So,
that's-
Oh,
sorry,
it
dude.
was this big.
it's
Wow.
huge.
That is huge.
Brittle
Huge
star
fish.
running away from him.
So off the northern Great Barrier Reef, they have Cod Hole, which is famous for potato cod, and they're about this size, and they congregate in one part of the Great Barrier Reef, the Cormorant Pass, I think it is, up near Lizard Island. Yeah, and of course it's a world famous place to visit scuba diving. You can go and dive with these giant potato cod, and it kind of reminds me of that place.
Yeah, I haven't dived there at all myself, Rob, but it's always been a place that's been on the bucket list.
Yeah, I've scuba dived there, and you can get face to face with cod as big as yourself.
Yeah.
It's incredible. Of how us biologists are talking about encountering them, and I hear fish and chips back there. People have different ideas of.
All right. We need now is a hammerhead shark to swim by.
Yes, exactly.
On the day will be complete.
That or a nice nautilus floating by.
Oh,
would
nautilus,
also be
yeah.
nice.
Yeah, it's about a metre long. Yeah.
I think you need to speak with your supervisor about getting you on a field trip so that you can do your own ground truthing.
I know, exactly. I think
These
I
guys
kinda
are going to
sold-
Heron all the
Yeah.
time. I'm sure they've got room for one more
I
student.
think I sold myself a bit short by just accepting to stay at home and study everything
Yes.
from the comfort of my home, whereas it would have been so great to actually get out there and
Yeah,
see
I
it
guess
for myself.
this year has not been the year for trying to do field work, though,
Exactly.
has it?
No, really not. But I do know some friends of mine who actually managed to get out to the Whitsundays or
Yeah.
a couple of reefs to get their own data. So, yeah. I think using remote sensing, that's the one thing you don't actually have to be there. You can gather the data
Well,
from the comfort of your home.
You need to be the person that's flying the drone.
Yes, exactly. That is true.
Yeah.
Especially with drone data, it's more of a
Very
in
nice
situ.
urchin.
Yes, it's
Big
beautiful.
urchin.
Yeah.
Oh, there we
Oh,
go.
that's another big one.
My giant cod encounter was on the President Coolidge in Vanuatu.
It's a big,
Yeah,
big
big
fish
old
there.
Boris, I think his name was by memory. It was a few years ago now. Resident potato cod, I think he was.
Yeah, I think I had my first big encounter with a potato cod at the Yongala,
Oh yeah,
just nearby Townsville. I think,
another dive that's on my bucket list. I haven't
it's
been there.
absolutely amazing. I've done dives, you know, kind of all over the shop, but the Yongala is one of those dives where it's almost a little bit too much
Yeah.
because every single second you see something new, so you can't actually concentrate on anything. You're just looking at one thing, and then there's another shark that comes by. There's just full of life. But a 40-minute dive is just really not enough to see everything.
No. It's reasonably deep too, isn't it? The Yongala.
Yeah, it's about
30
30 meters. It goes
Yeah.
down to about 30, and usually the current is quite strong. So
That was cool.
Alright, then Cody, you can come get it then.
Why?
Huh?
We're just gonna get to the top. Which one is that?
We're near the summit, aren't we?
We're near the summit.
Yeah. We've climbed the hill a bit quicker than we expected.
Well, we didn't do a great deal of sampling. I guess that's why,
No.
you know, sometimes we think we're not covering much distance, but the dive takes 8 hours
Yeah.
because we're doing so much sampling. But there
Yeah.
weren't that many sampling opportunities today. So we have gotten through a bit quicker than what we normally would.
So it was more of an exploration dive.
Yeah,
trying
for
to,
sure, definitely. Well, they all are,
Yeah.
actually. They're all
So we're
places.
pretty sure we've answered the question, what is it? What is this big knoll? And, you know, there were all kinds of ideas, but we're pretty sure we're looking at an ancient coral reef, which is now drowned. Drowned because the surrounding seafloor may have subsided and pushed it out of the shallow waters where coral reef grows, and now it's just sitting there as a big limestone hard rock, you know, resistant to weathering, and now just a home for some large gropers.
some beautiful red snappers that
You red
I'm lucky
snappers,
to
yeah.
see.
So the inset shows where we've been today. So we started on the deeper flanks of the ridge surrounding this knoll, and then we climbed up onto the steeper face. A big long traverse along the eastern face, up onto the crest, and we've basically walking our way up the summit now. So we're very close to near the top of it. This is all the mapping that we've done over the last week, revealing this really quite amazing, rugged seascape. The top of the knoll looks quite flat. Lots of sediment. Yeah, I think we can finish the dive and be pretty happy with what we've found.
Yeah, lots of fish, lots of sea stars.
Lots of urchins.
Yeah, lots of urchins.
Shrimp party.
Shrimp party. Shrimp. I want to see a shark.
Yeah. just at the top. Resident shark running around, standing its ground.
Cruising around,
Yeah.
scoping out his territory.
That would be a great prize after such a long trek up this mountain, basically.
Yeah, we've worked so hard to get here.
Oh, I'm sweating a little bit.
John.
Another one of these little edges where we tend to get these brisingid stars on
Sea stars, yeah.
the edges.
And if we're just looking at the inset, so that's us in the centre, but our destination over the next 2 weeks is to keep moving further north. So you can see the limit of where we've mapped over the last week, that's this grey-blue colour, just in this big embayment. And we're going to continue mapping further north towards the detached reefs, up towards the tip of Cape York, including around Brain Island, which is the world's largest green turtle rookery. So we're going to be mapping the deeper waters around all of these quite strange detached reefs. That's a real bird's eye view. That's Cape York, so we're probably looking at well over 300 kilometres of land there, and we're going to be working all along that very rugged shelf edge along the edge of the Great Barrier Reef.
You can see those detached reefs coming in the centre of the screen there now.
That's right. So they are very large reefs. They're sent out from the Great Barrier Reef, and we surmise that they're quite old. Like perhaps Miocene in origin, and we're going to be mapping around the edges of those, doing a full systematic survey up through Cape York. So this whole area we consider a frontier area of Australia. Cape York is very rarely visited by people. There's only scattered communities, indigenous communities, some mining camps, a few service towns, but it's essentially quite isolated, and certainly in these offshore waters, very few ships get up into these offshore Great Barrier Reef waters. So our goal here is to map all along the edge of Cape York and to find out what's there. And of course, this for us was quite a big discovery. This Tydeman Knoll, had no idea that it existed until we got here. So we now have mapped it in full 3D, and we have pretty good ideas to what's living on it. And when we bring the samples up to the surface, we'll be able to have a closer look at them. So the ship's been very efficient over the last 4 or 5 days, mapping quite a big area just offshore of Princess Charlotte Bay. It's a big embayment.
Beautiful urchin. Just a urchin. Yeah. Seen our fair share of urchins today.
Yeah,
really nice ones though.
Yeah, I think on that note we might call it, and that's it. Is that fish? Fish. All right, we'll look at these fish, and and then we might finish up the dive. We've reached the top of the summit of this knoll.
Oh, there's more in the background here.
Yeah.
It's a bit of a wander around here first before we call it. I love these sea star gardens. I look at all the edge here.
Yeah.
The whole lot is just covered.
Brings so much colour.
Yep. So I've learned about brisingid sea stars. I would have thought that they were feather stars, crinoids, but now I know. Yep. Sea stars.
Ah, that's a beautiful, beautiful picture.
Beautiful.
a few off. Quite steep. Are we over on the getting over on the western side then?
I'm not sure. No, we're still on the eastern side. Oh, there we go. Wow.
A bit hungry.
Yeah,
Actually, look at its tail. It's been
quite ragged.
Yeah, it looks a bit knocked around. This looks very old. This one.
Yeah, I noticed earlier when Gerald was flying by that cave, there's 3 or 4 of them that had those little black spots on their tail like that.
Yeah.
That tail fin looks really.
I wonder if it's
Quite chewed up.
I wonder if they're like bats when they go to lay eggs and maybe rub their tail on a bed or something.
Who knows? I have no idea.
Cat D is saying no Nautilus with a crying emoji.
No.
I feel the same.
Yes,
No,
I know.
today.
We'll have other opportunities. We're bound to see Nautilus. Right, we saw 2 Nautilus shells yesterday.
Yep. That was quite deep, wasn't
Yeah,
it?
it was. Yeah, would have been washed out.
Yeah.
We just have to get ourselves in the right depth zone, perhaps on the side of, right up against the edge of the Great Barrier Reef, and we might see some.
Antigonia. So we saw one of those earlier.
Boarfish.
Yeah.
Off he goes.
Let's chase that fish.
Yeah. Nooney's keeping track of the grouper tally. I think we're up to 10 so far. Do we have a grouperometer? Like we had a nautilometer on the last voyage?
Nautilusometer.
Yep.
Are we starting a grouperometer?
There we go.
Amazing.
I think we need to be up to 10 on the grouperometer. Oh. Oh, off the charts. Oh, 11. Woo. There you go. I think our maximum on the nautilometer was 7, wasn't it? Previously.
Oh,
That
okay,
is-
yeah,
Instead of
7
7,
probably
I think
one
it was-
time, but you know, one of those dives at Osprey, I think we counted up to 18.
Oh, wow.
You don't know if you're counting the same one,
Yeah.
you know, more than once. It's a nice wall. Let's see if there's anything hiding under there. real karst landscape, isn't it?
Yeah.
Completely different to the last dive that we did.
It really is.
Yeah, really. Yeah. So when we get to do these kind of mesophotic dives on the edge of the Great Barrier Reef, when we're much closer to the shelf edge, this is the sort of environment we'll be in. You know, it's very karst-like, karst caves and things like that.
Shrimp
Yeah,
party.
lots of shrimps.
Oh, that's a bit.
Oh,
He's
shrimp
gone in there
all on
to-
the right, up
Yeah.
on the right as well.
Kind of look like cleaner shrimp. Could be going in there for his cleaning session.
Oh, could be too.
Yeah.
Sorry, shrimps.
Oh, look at them all.
Whoa.
Oh, okay.
Bye, buddy.
Oh no. Chaos. Chaos in there. Whoa.
Saying goodbye to the camera.
So this tornado swept me.
Sorry, guys.
Just want to drop me out just a little bit more.
Come up into there.
Shrimp. More shrimp right along the edge.
Is there anything like the shallower, cleaner shrimp? My favorite part of a dive is always sticking in your hand in one of those holes and they come to your fingers and like eat off the dead skin off your nails. It's a free manicure.
Pretty game putting your hand in.
Exactly.
Yeah. You never know what's in there, like moray or something.
Yeah, exactly.
True.
You don't. There was a video that went viral of a man, an instructor who actually got his finger bitten off by a moray eel. I don't know if anyone has seen it. I know it went viral, but he was actually, I worked with him in Thailand and he showed me his finger and he's like, oh, do you recognize me? Oh, actually, you're famous.
Yeah, you're famous.
I think he was trying to feed a sausage to a moray eel to show some customers that were on a dive with him, and the moray eel just got a little bit too greedy and took everything, including the finger.
Yeah, but I mean, shrimp, they have these cleaning stations, don't they? And they'll just jump up onto a fish and
In their
walk all
mouth
over it,
and
pulling
their
off
gills
parasites and
Exactly.
loose scales and things.
So you get all the big groupers just floating there without really moving with their mouths open and all the shrimp going in and just having a feast.
Manip cam. One of the very many cameras that are
And look at
all
all
around the place.
the burrowing.
Sebastian.
When you look down on it, it's quite bioturbated. Lots of burrows. Another big urchin.
Snapper. Come on, hammerhead shark, where are you?
Out there.
Yep, somewhere out there. A tiger shark, big tiger shark.
Yeah, this is tiger
Tiger
shark
shark
country.
country. Okay, I don't know if this is an urban myth, but do you know if the moray eel finger guy got a toe reattached to it?
I'm not entirely sure, actually. I think there may have actually been a second guy that I heard had a toe reattached. This wasn't the guy that I worked with, but I have heard that myth as well.
think I'd rather just have one less finger than have a toe
Yeah.
on my hand.
Imagine trying to write with a toe.
Look like these blocks have broken away. Or they're just the eroded remnants.
Eroded around them.
Yeah, definitely the eroded remnants. Is there a roughy?
Okay, here's another story about a friend who lost a thumb and they attached a toe. Well, yeah, thumbs are pretty handy.
Yeah.
It'd be handy to have a substitute thumb.
Definitely.
Well, I worked with a guy in the Navy. He was a helicopter pilot, and he lost his thumb and had a thumb. Sorry, a toe attached.
to the thumb.
And he was quite a good helicopter pilot.
Yeah.
Yeah. It might not look the prettiest, but
No,
as
it
long
was a
as you
bit
have
messed up,
sort
but
of opposable
it
thumbs.
certainly worked.
Yeah.
A fringe of sea stars.
I'm just positioned all right around the edge. It must be the optimal position to be in to be feeding
Yeah.
on the current there.
The way the current's going.
Yeah.
The cup corollas are facing towards the current.
Yeah. Oarfish again.
I see a question from Dragonant about the drones. Yeah, so it's actually really interesting because you actually do get these hyperspectral sensors that are really, really small. So you can actually fit them on consumer grade drones. So any of the DJI Spark or anything that small, you get sensors that are maybe about 2-3 centimeters long. So really, really small, really light, and you can just attach it to your own drone, if anything, and you can actually get a lot of data out of that. So that's what makes, I guess, my project, what I'm so fascinated about is how not anyone, but pretty much anyone could actually get this technology and kind of conduct the same kind of project. Once I figure out what the methodology is, of course, then a lot of people can maybe,
It's one thing to be able to collect the data, but it's another thing to be able to interpret it. isn't it?
Exactly. So what I've been working on is trying to take this point data and trying to convert it into a more effective format for this analysis of spectral unmixing. So I've converted point data to raster data, and yeah, it's really interesting that you can use such a small sensor and get so much information from that.
So I've got a question to you there, Brody and Chris. Have you guys ever put infrared cameras on, or red light cameras on SuBastian?
Infrared, yeah, we put, what was the one we put down?
We used a yellow lensed camera to see what sharks see with some ultraviolet lights, and there was a few scientists that were studying how sharks can see certain depths and what they actually see. So not really infrared.
Yeah, the UV light had some different rock and some different features would glow.
Yeah, will glow a certain way, and that's how they thought the sharks could sense their location and what's out there for maybe putting their eggs or whatever down. But no infrared.
You ever put red light cameras out there at all? No? So it's all white, yeah.
Lights. Now lights, we did have a red cap on a few lights for siphonophores when we're in the White Shark Cafe in the middle Pacific. We did a few red lights to look at siphonophores with a different color to see their different stomachs.
I was not here for
Oh, you
that.
weren't?
I was
Before your time.
I was in Canada.
Yeah.
What about you in Canada?
What was that?
I might sign off.
You want to call it?
Yeah.
All right.
I think we might come to the end of our dive and reach the
Yeah,
summit.
we've had a good look around here, and it's been lots of fun. It's been interesting. I think the gropers were the highlight.
Gropers and shrimp parties.
Yeah.
Yeah.
But look, thank you very much for everyone joining us. We're going to ascend now, bring the SuBastian up to the surface, probably about half an hour worth of ascent, and then we'll pull our samples out and do the preservation. But this was our fifth dive of this expedition. This is the northern depths of the Great Barrier Reef expedition, where we're really focusing on the quite remote and frontier Cape York region of the far northern Great Barrier Reef. So we intend to do as many ROV dives as we can, weather permitting. So perhaps every second or third day we'll try and get an ROV dive in, and we'd really welcome everybody from ashore to join us for these live dives. So we'll say goodbye and catch you on the next ROV dive. So this is ROV Falkor out.